Good morning, everyone. Welcome to the 10th World Conference on Pharmaceutical Science and Drug Manufacturing on the theme An Integrative and Holistic Approach Towards Pharma Sciences, organized by APR, Association for Pharmaceutical Research, and BioLeaks Association. I welcome you all for the day 2 of the conference. Hope the day 1 has been successfully done, and we are hoping for the same for the day 2 also. I welcome all the participants, teachers, lecturers from the colleges of various pharma colleges, and also our keynote speakers. I would like to welcome our keynote speakers of the day 2. Professor Dr. Sadia Sultana, Head of Biotransformation Research Group under RMC UITM, Faculty of Pharmacy, University Technology, MARA, Malaysia. We welcome you, ma'am. Mr. Ankit Sharma, Research Manager, Sun Pharmaceutical Industries Limited, Haryana, India. Dr. Sundaram Ramachandran, Chief Scientific Officer, Injex Lab, Karnataka, India. I welcome all the keynote speakers and heading on to the sessions. Once again, I'd like to welcome you all for this Global Pharma event. I'd like to welcome our first keynote speaker of the day, Professor Dr. Sadia Sultana. She is an Associate Professor of Pharmaceutical Chemistry in the Department of Pharmacology and Chemistry at the Faculty of Pharmacy, University Technology, MARA, UITM, Malaysia. She is appointed as lecturer in the Faculty of Pharmacy, Shah Alam, as a Fellow of Utar R. Rahman University of Natural Product Discovery. She is having more than 20 years of experience in the field of natural product research. Her area of expertise includes biotransformation of exogenous substrates using microorganism and plant tissue culture technique, exploring bioactive secondary microorganism and exploring bioactive secondary metabolites from the plant, soil, and marine endophytic fungi. Modern MNR streptosophic approaches in the secondary metabolites biological screening using antimicrobial assay. She has over 70 research publications in top international journals Q1 and Q2, including high impact factor journals like Pharmaceuticals, MDPI, European Journal of Medical Chemistry, etc. She is also an editor of the book Fungal Phytogenicity. Her current research is based on the utilization of modern NMR streptoscopic approaches in secondary metabolites, biotransformation of an exogenous substrates, and synthesis and characterization of bioactive medicinal derivatives using combinatorial chemistry. For her research work, she has been awarded several national grants as a principal investigator, including eScience, Ministry of Science and Technology, FRGS, ERGS, PSI, DANA Fund, and REI. Her research findings are also recognized as several conferences, competitions, and exhibitions, and awarded with several awards like Best Poster Awards, IDGX Gold, IDGX Silver, and IDGX Bronze Awards. Over to you, ma'am, for the session.
Hi, hello. Thanks a lot for the organizer for such a nice, overwhelming introduction and give me an opportunity to share some of the part of my research work over here. Today, I'm just sharing one of my PhD students. She has currently graduated from as a PhD, and she has a chance to have some attachment work at Norway as well. And she went there and she has collected herself some samples and isolated some endophytes. And you can see her name over here as well, Fatma BB, and her another co-supervisor of Kalavati. So first of all, before I just show you some of her interesting results, maybe some of you don't know about what are endophytes. So endophytes are isolated from the plants, and they are the microorganisms. They are living inside the tissues of plants in organs like leaves, stems, roots, and flower as well. So we just isolate these endophytes from the plants, and then we grow them on a large scale. First, we have to be confirmed they are pure, and then we isolate different types of natural products. So we all know we okay. So we all know the natural products as a main substances, and they are the model molecules for the discovery and the validation of drug targets. And about 60% of the drugs are derived directly from the natural products, and the rest from the synthetic means. So, according to Newman and Craig researchers, scientists, they have reported a significant numbers are isolated from the natural products by microbes. Some are from plants, some from tissue culture, but 60% mostly significant numbers from about from the 60% are from the microbes, and they are mostly from the endophytes as well, microbes living inside the plants. So what are endophytes? Endophytes, as I told you, these are the microorganisms, and they are find inside the internal tissues of plants and without causing any infections or disease. Actually, they are useful for the plants. They don't harm the plants. And we have different types of endophytes. We can have from the tropical or temperate or rainforest zone, such as Malaysia is come under the rainforest, and it has a great biodiversity, and they are rich in endophytes. And endophytes from the extreme environments like Arctic or Antarctic zone as well. They all are promising natural products resources as well. So the potential source of this biologically active secondary metabolites have remarkable structures as well. As we know, they can be polyketides, they can be alkaloids, they can be terpenoids, peptides, and steroids as well. So how we have driven towards this one to do this type of research. We all know the current very situation we have antimicrobial resistance. We are all are facing this antimicrobial resistance, and this is increasingly seriously, and is a threat to global public health as well. Natural products is unlimited, and we have a majority of the world's diversities, and we have to explore, and all this remains to be explored. And natural products from this endophytic microbes can be used for the treatment, and then they can be a drug resistance bacterial infections as well. Endophytes, this express have to go first for the antibacterial activity, and it can be a new chemical entities as well. And they have potent and strong activities. Endophytes from the plants, here we are discussing from two different regions. My student, she worked on two different regions. She worked on tropical and Arctic zone. So tropicals, she collected samples from our own university campus in Pujalam, and from the extremophile, she has collected from the Arctic zone, North Pole zone. Okay, so if we see briefly the methodology, how we did, we have the collection of some plants, and then we have isolated the endophytes from the plants using agar, and after this we have culture, we have incubate for few days, and then when we observe some colonies, we have separated, because for one samples, if we take small piece of, for example, a leaf on agar plates, and when you incubate for a few days, you can see different types of colonies. So we have to separate each type of colony in different plates, and later we have to grow for some further more days, and then we have to confirm this one is pure, and then this one we can confirm by using the microscopes. So after this culturing extractions on small scale initially, and then later on up large scale, we have initially we select on a small scale some strains on the basis of biological activity, and which we can see is really pure when we make it sure, right? And then after this we have done HPLC profiling. HPLC profiling help us to see the peaks, significant peaks, which are easy to isolate and which have a difference and good resolution as well. Then after once we have done on large scale, we do explosive fractionation, isolation, and purification of our metabolites. And parallel, we are also doing the morphological examinations of our isolated fungi. After examination, identifications, and then for structural elucidation, once we have pure compound, or initially on extract basis, later on pure compound, we have done the biological assay as well. So for the collection from the equatorial, we have done from the equatorial and Arctic plants. From the equatorial, we have collection area is the Manharba. We have from the Manharba, UITM Punjalam Biological Research inside our campus. We have collected 3 rubiaceous species, and for each species, we have petals, leaves, stems, and roots as organ of plants. And then we have sent it for the identification for our forest department stream for the identification of plant. For the second one, we have collected from the Arctic plants. Arctic plants, the collection area is the North Pole, Arctic, and we have 10 species we have collected, and including the organs, petals, leaves, and stems. And its identification is done by Nobel botanist Dr. B.P. Edison. Okay. So for equatorial plants, we have 3 plants are identified as Ancaria. And from the first plant, we have isolated 21 endophytes from different parts, organs of Ancaria. From the second plant B, we have isolated 20 endophytes. And from the third one, we have isolated 13 endophytes. Overall, 54 endophytes are isolated from 3 plants, equatorial plants, collected from the Punjalam campus. And they all are grown on 28 degrees centigrade, all the endophytes. You can see the pictures of plants as well. Okay. So how we have done the isolation of the endophytes. So from the Arctic, we have total of this 13. And we have grown this Arctic plant on 2 different temperatures. One as a cyclophilic fungi as 10 degrees centigrade, and as a cyclotropic fungi at 28 degrees centigrade. Because we want to see difference between once it grows on their own environment temperature, like 30. As you all know, the extremophiles can be grown from the -20 until the 20 degrees centigrade. So we have, for our lab setup, we have grown it on 10 degrees centigrade and 28 centigrade as a cyclotropic. So for example, the first plant, the papaya, we can see we have isolated 3 fungi, and as a cyclotropic, we have isolated 2 endophytes. So overall, from the psychrophilic, we are able to isolate 13 and fungi, and from the psychotrophic, 10 fungi. These are the pictures of the some plants, small plants we have collected from the Norway Arctic zone. So initially we have checked because we don't go the randomly to work on all. We just in order to make it specific, we just go screening. So we have done antimicrobial assay using MTT assay. And these are the microorganisms: E. coli, Staphylococcus aureus, Pseudomonas. We have used this, and Candida albicans used as organism for screening base. So the screening results have shown here. So for different microorganisms, we use pathogen E. coli, E. faecium, Pseudomonas aeruginosa, Staphylococcus aureus, and Candida albicans. And the number of active extract from the equatorial endophyte, but we have two equatorial and Arctic endophyte. From the, you can see here from the, for the E. coli, out of 20, 11 are shown to be active. For same for the Pseudomonas aeruginosa, out of 22, 12 shows as an active. Same for the Arctic one, from out of 19, 6 should want to be active. So these are the number of active extract from the equatorials out of 54 extracts, and these are the number of active extracts from the Arctic endophytes from total out of 31 extracts, because we have come out with the 31 extracts. So out of all of them, we have selected 4 crude extracts as a PhD objective. She has selected only 4 crude extracts that have exhibited a broad spectrum of activity, and this is selected for further investigation. So then she has gone for MIC, and you can see the MIC of different extracts from different plants. So this one is, these 3 are from tropical region, Punjab forest, and this E1 PB is from the Arctic one. And these are the standard drugs as well. So this one E1 PB1 was selected. This extract was selected from the Arctic, and 3 extract was selected from the tropical: TH1P45 extract, TH2S14 based on this MIC, and TH3R30 extract based on this MIC activity. Sorry. So then we have moved forward for the chemical investigation. After we have finalized the selections, then we have moved on further investigation for the selected 4 extract. So for the fungal identification, we have done morphological and molecular examination. We have SEM facility in our institute. So first on the DNA, and then submitted to gene bank as well. And then we have sent for fungal identification for SEM as well. I will show in later slide as well. And further, we have done the HPLC profiling, and then fractionation, isolation. For we use PrEP and semi-PrEP HP and recycling HPLC using DAD and ELSD detector, because some can be UV inactive as well. So for the identifications of bioactive metabolites, we have used high techniques like MS, NMR, UV, IR, and ECD. And we have compare our results with the literature as well in order to find either new or known compounds. Okay. So for the morphological examination of the fungi, we have gone to microscopic practice first. We have seen morphological, how it looks like in PDA, because we have a good collaboration with New Zealand professor, Tony Cole. Most of the time he was with us as a visiting professor from the New Zealand. So we have found out on the morphologically initial identification of our fungi. Then for microscopic characters, we have done using the microscope after scanning using lactophenol. And then finally we send it for SCM. This facility we have our institute, SCM microscopic. So this is the first one, extract TH1 P45. You can see how it's look like morphological on the PDA. And it's after identification, it was found out to be Nigrospora rhiza, and it submitted to gene bank. It is found to 99% of similarity. So this one is under microscope. We have optical microscopy. So you can see here this Candidiophore and hypha of this microorganism, and this is we have done on SEM, and it's further confirm the identification of this microorganisms. Okay, this is a second one, and this one you can see this is TH2S14. It also submitted for gene bank, and it is 99% similarity. So this one under optical microscopy, again you can see the spores and filaments. If it's normal, if we have spores, very easy to identify based on the morphology of the spores, easy to identify. But we have further gone for the extraction of DNA, submission to gene bank, and other like SCM. So in SCM, you can see very nice magnifications of this microorganisms. You can see the filamentous ring look here. So it also helps for the identification. And again, if you see the spores, so this is Conidia-4 and alpha and beta Canidia. This also helps for the identification of this fungi. The third one is THTR30. This is Trichodherma, and it also have 99% similarity. So this one is also, you can see on the optical microscopy. And further, this is a very nice capture of in under SEM. Sphalytes, you can see is a mass of conidia and phthalates. Very obvious here. You can see for this is magnifying over here as well. This is this extract is selected from the Arctic, and this one again on optical microscopy you can see it has hyphia and conidia, and further an SEM again. This is spores, and these are very obvious hyphia over here. So for fractionations from the HPLC, for the first one, we have further fractionate our subfraction of the first one. We have F1 until F9. And from third fraction, we are able to have one compound from F4 also resemble one compound. So overall, you can see here we have 5 compound from first extract. So here further on HPLC, it's very HPLC profile. So we have overall 5 compound from our first fractions. So this is very clear HPLC profile. You can see here we have focus more on the compound which are on high yield, and the small one we have this one can be done later, but for the time being we have just focused on the peaks which have a high intensity and high yield, and they are already identified as no compounds. So this is the second one. From the second one, you can see we have 2 compounds. The one is 30.2 mg, and another one is 6.6 mg.
Translated comment in Hindi, you understand how that? And then because I watch one.
These two compounds are belong to cytochalasin family, and this one also isolated. And this you can see these two compounds are in high yield. So based on that, we can also can say we can use this. If these compounds are very active, we can use these two compounds. We can produce this compound with the help of this fungi, because this is most of the time this is reproducible, and we can isolate this two compound from this fungi. And these are, if this is the selectivity, we can this is in high concentrations. So this is the beauty of this work as well. Okay, the third one is we have TH3 R30. And from this, we have three compounds again. And if you see the HPLC profile, again, you can see these two compounds are in these two are the major compound for this extract. Okay. This is for the Arctic one. So as you see in my title, I focus on the Arctic one because sometimes this is more interesting, and this is the rare work the people are doing. So because it's not easy to go there, collect a sample, and then it survives, and we have to take care, and then we have to grow here the fungi and isolate the compounds. So from this Arctic extract E1PB, and you can see we have major components here. So overall, here we can see 5 major components here. And overall, the extracts was 638 mg. And from this, we have different compound in. Some are enough and some are not enough yield. Okay, so this is from the Arctic specific profile from the Arctic extract. So this is the major compound. This one also major, and this one also, and this one, this is the same extract, and these three are the new polycyclic polyketides we are able to isolate. And this is the novel compound as read from this Arctic zone. Okay, so this is the NMR spectra for each compound. You can see one here. So you can see here the NMR spectra. This is 2 metacoupled aromatic protons, because we have aromatic proton as well. Okay, so this is an C13 for this compound. This is a second compound. This is an NMR for the second compound. This is a C13 for the second compound. This is the third one, which is the novel compound. This is the NMR for the third compound. Actually, the structural elucidation of this compound was very challenging for us, and but we are able, and we have already, my student has already published this paper in JNP as well. So this compound was challenged for us, and we took help from mass experts, and we have deduced the structure on the basis of mass fragmentation of this compound. So this is the mass fragmentation pathway for this novel compound, which help us to deduce the structure of this novel challenging compound. So these are all the results from the mass fragmentation. So 5 compounds are isolated from the E1PB extract subjected to the biological screening, and we have done in vitro, because first we just use the in vitro, and then we, if any good activity, then if enough amount, then we can go for the in vivo activity. So we have done antimicrobial assay, but unluckily none of them have shown any significant activity. But this is the limitation. If we can go for other activities as well, but due to the limited amount of the compound, we just able to do only this activity. But this one can give have idea for we can regrow this compound and can have produced in high yield, and we can go for other activity as well. But it gives some minor, less potent, but still some activity they have. Okay, so this is the activity of these 5 compounds. We have screening for this one. So you can see there is some cells protected from the induced cytotoxicity as well. So based on that, we actually we can just conclude was to this presence of bacterial metabolites of this endophyte. So we can say we have novel compound, novel secondary metabolites from this Arctic extremophile samples as well. And overall, 54 endophytes were isolated, 3 from 3 WCPSDM plant and 31 isolated from 10 different civil plants from Arctic. And we have done HPLC profiles and was selected based on the screening of antimicrobial assay. And 4 extracts was selected out of this endophytes and for further studies. Okay, so overall, total 16 compounds were identified from this selected extract. It include the 11 known metabolites and 5 new compounds, and 5 known compounds from Nigrospora arises, and 2 known compounds from it's already mentioned here, cytokalisine, and 2 more compound from the trichoderma, and 2 from Squalia species as well. So we can use this one for other biological assay because we just use this limited. And one thing which is very interesting, which we are also doing here, we are using OSNEC approach as well. One strain, many compounds. What we can do for the same extracts, for same fungi, we can use different, because this time we use one type of media, we can use different media, and we can use different elicitors, different minerals, different trace elements. We can add in the media and can see what, because you can see here some of the compounds are not in sufficient, not in efficient yield. So with the help of elicitor, we can enhance the yield of our compound of interest as well. And this one we are doing, but we haven't tested for this specific Arctic endophyte, but we are already doing this one as well in our lab. And in this way, we can have more compound, more different type of compound, and the existing compound in high yield as well. Okay. So before I have one video. I will show you a small video after this one, what we are doing here. Just our all achievements, because I'm also the head of, previously we have RAG, now last year, our based on our achievement, our group is upgraded to research group as well, and I'm heading this group for I think for last 7 to 8 years. I'm heading this group, and I also would like to acknowledge my internal RMC grant, and this grant, this couple grant, awarded to me based on this achievements. And I'm also very thankful our Dean of Faculty of Pharmacy, Dr. Abubakar Abdul Majid. He's really very motivated and cooperative for all of us. And my Faculty of Pharmacy, I'm working here for last 18 years. And thanks. Before I just stop, I want to share my video with you, just few minutes, 2-3 minutes only.
Sure, ma'am, sure.
Have I already stopped sharing?
No, ma'am, you have not yet shared, ma'am.
Still sharing. Sorry, my
I hope there's a network issue.
yeah, exactly. So
Image of yours is glitched in here.
you can hear me?
Yes, I can hear you, ma'am.
Oh, the Zoom meeting is not responding. Give me a few minutes, otherwise I stop here. Just give me a few minutes more.
Sure, ma'am, sure. Ma'am? Sadia, ma'am, are you there? Ma'am, are you there? Ma'am? Ma'am, are you there, ma'am? I think Sadia, ma'am. I think Sadia, ma'am, is trying to. I think Sadia, ma'am, is trying to join. We'll wait for a few minutes and see.
Presentation started. No.
Okay, I hope she's facing some internet issues. We'll move on with the next presenter. Can anyone respond? Am I audible?
Yeah, Shashmita, you are audible, but we can wait for 2, 3 more minutes. No issues from my side.
Okay, sir. Okay, we'll wait for a few minutes. Whenever you're ready, please let me know. Ma'am has told we can move with the next speaker.
Okay, just wait for 2-3 minutes.
Sure, sir, sure.
Actually, I'm driving and I'll.
Is it possible we'll move with Dr. Sundaram next to his you can presence, sir? Will that be possible? Mr. Ankit? Okay, Mr. Ankit will join. You can join your presentation next to Dr. Sundaram.
Okay,
We'll
no
move on
issues.
to the. Yes, sir. Yes.
Okay, no issues.
Okay, okay, sir. We'll move on. Okay. I'd like to welcome Dr. Sundaram Ramachandran. He has completed his PhD in 2007. His research interests during PhD were activity guided fractionation, isolation, and purification of marker compounds, molecular biology, and in vitro pharmacology. Dr. Sundaram Ramachandran worked for Himalaya Wellness Company as research associate in the year of 1994. He has worked at various positions in the company till 2022 and handled activities like method development, process development, activities development, formulation development, and technology. Dr. Sundaram Ramachandran have established strong technology transfer protocols from R&D to commercial production. He interacted with various regulatory agencies for product registration over the 100 countries and also conducted workshops for regulatory bodies like Oman, Bahrain, etc. to give full perspective about the natural products. In 2023, he has joined as a Chief Scientific Officer in Ignex Lab Bangalore. He is currently handling research and development activities of herbal extracts, naturoceuticals, functional foods and cosmetic products with herbal activities. Dr. Sundaram Ramachandran is an expert panel member in United States of Pharmacophobia, USP, dietary supplements and herbal medicine, South Asia, expert panel and contributed several monographs to the USP on behalf of earlier organization. He works very closely with BIS as an expert panel member in cosmetic sectional committee and has given various inputs in Ayush Pharmacophile committee as an expert panel member. Dr. Sundaram Ramachandran have more than 50 original research publications to his credit and he has delivered more than 30 lectures in national and international conferences and workshops. Over to you, sir.
Very good morning, ma'am. Thanks for the kind introduction. I will share my screen.
Yes, sir.
Can you let me know if it is visible?
Yes, sir. Your screen is visible, but kindly can you make it as a full screen?
Yeah, I made it now.
Yes, sir. It is visible. Kindly forward.
Yeah.
Thank you, sir.
So at the outset, I would like to thank the organizers of this 10th World Conference for giving me this opportunity. I was thinking about the topic which I can choose for this conference, and unfortunately, I don't know anything about other than the natural products. So I took the natural products as one of the interest to present here. So I just would like to title my presentation as Inspired by Nature, as we get inspirations from nature in many aspects in our life. And similarly, the modern medicine also got a lot of inspiration from the natural ingredients and natural products. So because of that, I took this topic. So if we all keenly see, 1828 was the first synthetic organic compound produced, that is urea. And after that, the synthetic organic chemistry evolved and a lot of synthetic drugs came into the picture and the limelight in the treatment of human diseases and disorders. Till that 18th century, there was no synthetic medicine available, and every country or every continent of this globe was fairly dependent on the natural sources, may it be plants or animals or minerals. So humans were exploring different kind of these ingredients as medicines for curing their diseases or disorders. So if you see, there are various traditions, East Asian medicine. If you talk about, there are Chinese, Japanese, Korean, Mongolian, and Tibetan medicine. And in South Asia, we talk about Ayurveda, Siddha, Thai, Sri Lankan medicines. And same way in Mediterranean, we have medicines like Iranian, Roman, Unani, and all that. Same way, American also had a lot of traditional medicines, and Africans also had a lot of traditional medicines. If you keenly see how these medicines were invented or discovered by our ancestors without even having any laboratory or any instrument or any animal models, it is very inspiring for us. So they all observed the nature, and they also observed the animals, how they behave, how they consume different plants, and how they make use of different plants as either food or curing some of the diseases. Man also experimented a lot of plants on himself and identified what is potentially a medicine, or what is potentially a food, or what is a toxic plant or something like that. So a lot of sacrifices were done in this process, but a lot of medicines were evolved during this process. So in India, if you see, Ayurveda had given a lot of classical medicines. Mainly, they are all combination of different medicinal plants for curing different diseases or disorders. So though it was not scientifically validated as per the current scientific practices, most of these formulations still work, and still many people are getting benefit out of it. If you see, even these medicines have thrown a lot of molecules to the modern medicine. If you see, the principles of modern medicine is to validate the efficacy of the product, and by the questions like what is working, why is working, and how is working. So we talk about drug mechanisms of actions, the structure activity relationships, then ADME, the absorption, distribution, metabolism, and excretion. All these things are major considerations in the development of modern medicine. So a well-defined molecule from a natural product with all these properties actually eventually came as a mainstream medicine in the current systems. So you talk about morphine. As early as 1804, we had isolated morphine, and characterize the structure, and the mechanisms were identified and used as a medicine. Same way, acetyl salicylic acid was invented. The discovery of penicillin gave a major breakthrough in the antibiotic research. And same way, tubocurarine became an anesthetic agent, and Vinca alkaloids became a treatment for cancer. Same way, Taxol also became a major popular drug, and artemisinin became an anti-malarial drug, captopril. All these drugs were invented from the natural ingredients and natural molecules. So this inspiration has really given lot of insights into the modern drug discovery also. If you see, even post-COVID, many people have realized that synthetic medicines are not going to be giving solutions for all the problems. So Now people started depending. There's a lot of disturbance. Can people mute if they are not speaking?
सारी जूम पे है ना, ये जो सारी यूट्यूब पे हो रहा है, क्या फायदा कर हमें यूट्यूब से सुन लेते हैं?
Hello audience, can you mute your mics please? Yeah. During COVID times, all of us have used some herbal medicine or the other to improve our immunity. This all we have practically experienced. So if you see the herbal medicines market, this is the picture where we see the 2020 picture and 2030 picture. There are 5 different categories, 6 different categories. So the major category is the health supplements and medicinal teas and nutritive jams and cosmeceuticals. These are all the major areas. So if you see the fastest growing segment in this, the whole herbal industry is the medicated teas, that is fastest growing. And in terms of market values, if you see, herbal supplements are forming the biggest chunk of the market. So if you see the developed countries, many people have now started consuming a lot of herbal supplements to improve their health and well-being. This is visibly seen in major countries. If you see another chart where from 2022 to 29, there is a projection given, the market is likely to grow to 177.65 billion by 2029. And if you see, currently we are standing at 23. From 23 to 29, we are expecting a growth of 3 times, more than 3 times in the market share of the herbal medicines. If you see North America, Europe, Asia Pacific, South America, Middle East and Asia, Middle East and Africa, all are equally distributed in the market share. It is not that one continent or one country is consuming the herbal medicines, other is not consuming. So every country is equally consuming the herbal ingredients or herbal medicines. So this is the overall background about the natural products industry and natural products market. So I just try to do a simple SWOT analysis for the herbal medicines and herbal ingredients. So what are our strengths and what are our weaknesses? What are the opportunities and what are the threats? So if you see, in terms of strength, India is having a rich biodiversity of a lot of medicinal plants and a lot of traditional wisdom. As I spoke, a lot of classical medicine still working very good in the human diseases. And this traditional wisdom is helping to modernize these medicines. And they are relatively safe when compared to the synthetic medicines that we cannot ignore. And the same way, what are the weaknesses? If we talk about the weaknesses, still, though there are a lot of scientific advances, still these herbal ingredients or herbal medicines are poorly studied. And a lot of seasonal and geographical variations in the collections, in the ingredient sources. So that is impacting the quality of these medicines. So these areas need to be strengthened to make these herbal ingredients popular and global. And looking at the opportunities, there are improving global awareness. Then every country, as we spoke, every continent is consuming the herbal ingredients and herbal medicines. So there is an improving global awareness about these herbal ingredients, and they are widely accepted. And the improvement in the science and technology is broadly helping the research point of view. So from the research point of view, we can validate our herbal medicines doing a lot of research and the structure activity relationships, those kind of activities. And coming to the threats, the supply chain is one of the major constraints. As the demand is growing exponentially, the supply also has to meet with the demand, and that actually is a big challenge. A lot of cultivation and activities like farming is to be improved in the herbal medicines area. And sustainability is one of the major threats. So we keep consuming from the nature and we are not replenishing them. That is biggest problem for the herbal industry. And the regulations are not harmonized like allopathic medicines. Allopathy, we talk about ICH guidelines. Once we follow ICH guidelines, we will be able to take the products throughout the globe. But in terms of herbal medicines, the regulations are not completely harmonized. Some countries call them as a dietary supplement. Some countries call them as herbal medicine. Some countries call them as phytopharmaceuticals. Some countries call them as a natural foods and all that. So the regulations keep changing from country to country. So it becomes very, very challenging for people to follow one regulation and market throughout the globe. These are the challenges or threats for the herbal drug industry. But still, we can overcome these challenges and make these herbal medicines globally popular. So I would like to emphasize this in the herbal medicines. Herbal medicines, when we talk about, is a concoction of various molecules. It is not a single molecule. It is a concoction of various molecules. If you see a photosynthetic pathway, each herb produces a lot of phytochemicals constituents like phenolic acids, phenylpropanes, stilbenes, flavonoids, tannins, lignans, and all that. So if you look at very carefully, the plants are synthesizing these compounds for their own survival and safeguarding themselves from various environmental stress and infections and all that. So these metabolites or secondary metabolites, we are using for our benefits as medicines. So we need to have in-depth understanding about each plant and each metabolites in each plant and see how they will be useful for our benefits. So that is why we need to employ modern tools in terms of chemistry and in terms of biology. So a lot of advancements are happening in chromatography, and we can separate, purify, isolate, characterize these compounds. So whether it is a single major molecule, we can separate, or multiple component mixture, we can separate, and we can do a chemical fingerprinting. So these are all very in terms of Dr. Rajesh, can I request you to mute the mic? Thank you. Yeah. So in terms of spectroscopic techniques, so we use NMR, instruments, and we use near IR, we use Raman spectroscopy, all these spectroscopic techniques for characterization of these isolated herbal constituents. In terms of biological evaluation, now people are using a lot of bioassay studies, the in vitro assays, in vivo assay, and different kind of models for evaluation of the biological activity. And the important point is the integration between chemical and biological evaluation. So only when we integrate these two things, then we will be able to get a clear science about the actual efficacy of the medicine. So we need to integrate this data by various techniques and then understand the mechanisms of action of these molecules. So this in early 90s, this became a popular tool in herbal research, bioactivity guided fractionation. Each herb is having so many molecules, so make a crude extract and do different chromatographic techniques like flash chromatography or preparative chromatography, and then I analyze each fraction for the biological activity using ELISA based methods, then identify the most active fraction, and then purify the compounds using chromatographic techniques, and then do the structural elucidation of these molecules. So this became an actual eye-opener for the herbal research, and many people have isolated many compounds, characterized them, and used these molecules for standardizing the extracts, and then take these extracts globally into various markets by proper scientific validation. So another approach is when we talk about an unknown molecule and then identifying the property of those molecules by this discovery based approaches, it really takes a long time. Like it takes 10 to 15 years to bring out a medicine which cost more than $1 billion US. For example, any synthetic molecule, is discovered in this way. Target identification is done, and then the lead identification is done, lead optimization, preclinical study, clinical study, and final documentation and FDA approvals. So this is very lengthy process. When we talk about herbal medicines, we can follow a reverse pharmacology approach, where we can use the traditional knowledge database, and then using this traditional knowledge, we can actually reinvent these medicines. This extract, which is mentioned in the classical Ayurvedic text or classical traditional medicine textbooks, then we can straight away go for a preclinical study and validate the efficacy and mechanism of action, and then we can take them to large-scale clinical trials and then go for approval to various agencies. Similarly, want to make a purified fraction from these traditional medicines. Still, it is possible. We can fractionate and identify the active molecules, and then straight away go for preclinical studies and clinical studies and go for FDA approval. So, if you look at the Indian government also is encouraging this approach. They have come out with guidelines on phytopharmaceuticals, where we can take a known medicinal plant and then characterize 4 different active marker compounds from that plant, and then straight away we can take them to preclinical studies and clinical studies, and we can file in our Indian FDA as a medicine. So this approach is much shorter. It takes only 5 to 6 years, and we can develop a product in to $20 million. So this approach, many companies are now following. Even well-reputed global pharma majors also, they are coming into this area. They are coming into this pharmaceutical area, and they are developing various phytopharmaceutical products. So this I just wanted to emphasize here. So if we look at this target-based approach, as we spoke, there is a herb and there is a molecule, and that works on particular mechanism. So herb is the holistic thing where it contains so many molecules, and we make an extract out of it, which may be containing several molecules, but still it is not as pure as a molecule. Then we can fractionate them, and then we can convert into the molecule. So in my experience, I have studied not all the herbs work in the same way when we purify them, molecular level. So herb is working, extract is working, fraction is working, but the molecule is not working. This kind of situations I have practically seen in my career. And when we try to understand why a molecule is not working, but the fraction is working, then we understood at the fraction level, it has multiple molecules, which actually acts synergistically and then give the efficacy. So we need to also understand, when we are doing a research in herbal medicines, at what level we have to stop, whether we have to use as a crude herb, or we have to extract them, or we have to fractionate them, or we have to still purify them. So at what level we have to stop, also we have to carefully evaluate, otherwise we will be losing the activity, and we will end up doing more and more purifications. So if you see, even there is a cost involved on every step. So if you talk about a plant material, there is a dried powder, then non-standardized extract, then there is a standardized extract. So at every activity, there is a value addition, there is a cost involved. So the moment you purify more, you are going to actually spend more money, and your cost of the product is going to increase. So when in order to purify so much, then you have to stop at some level, maybe at this level or at this level, we have to stop, so that you can save the cost also, and we can also bring a therapeutic product without compromising the efficacy of the product. So this is very important to understand in the herbal drug research. So here, coming back to this molecule level or fraction level research, this network pharmacology approach is now currently being adopted by many people who are working in the herbal drug research. So now there is an understanding, like not always one molecule works, but there is a combination of different molecules which gives a synergistic activity. So in traditional pharmacology also, drugs are often used in isolation, focusing on their interaction with the specific target proteins or pathways. We all studied in our college that a beta blocker gives the activity by just binding with the beta receptor. So now people have understood that it is not the beta blocker is only acting on the beta receptor. It is also acting on so many other targets, and there is a network of different proteins and different genes. So this network pharmacology takes more holistic approach by considering the interconnectedness of biological systems and multiple targets and multiple pathways in the drug research. So the key principle of network pharmacology is that diseases are caused by disruptions in the complex biological networks. There are biological networks, and there is a disruption in the biological network that only causing the disease. So this also coincides with the principle of traditional medicines like Ayurveda. So Ayurveda says humans are having imbalance. When there is an imbalance in the metabolic status, there is a disease. So they classify the body as Vata, Pitta, and Kapha. And when there is an imbalance between these three major forces, there is a disease. So this modern principle also tallying with those olden traditional principles that there is an imbalance in the biological networks that is causing the disease. By studying these interactions between the drugs, these networks, the researchers can gain insights into the underlying mechanisms of diseases and also find out new therapeutic targets. Network pharmacology utilizes various computational and experimental techniques to analyze the model interactions between drugs, target proteins, the biological networks. These techniques also include network analysis, data mining, bioinformatics, molecular docking, and simulation modeling. So as the technology is improving in terms of information and technology, so a lot of such tools are coming into the picture. A lot of computer aided design and computer aided models are available to map this network pharmacology. So if you see here, traditional herbal medicine discovery, we always were taking a cue from Ayurveda and then identify their pharmacological activity, then also isolate the molecules and characterize them using different spectroscopic and chromatographic techniques, and then find the toxicity of these molecules, and then developing the product. This is how we were doing in the traditional pharmacological approach. But with this network-based pharmacology, so the metabolomics profiling is becoming more important. We need to understand target network modules, functional modules, and metabolic pathways and all that. So a lot of publications are coming into picture particularly from traditional Chinese medicine. People are actually trying to understand the medicinal plant and the disease in depth using this network pharmacology technique. So if you see, this is from one of the published paper. People have used a medicinal plant for particular disease, and they try to do the data mining from the databases or literature or lab-based experiments. They find what are the components in the plant, and then they also identify the pathways of the disease, and then they construct this kind of pathways, and then they validate this using these computer aided models, and then finally they go for laboratory experiments and prove those activities in silico, followed by the in vitro or in vivo models. So this is how the networks are created. The medicinal herb is fully understood for the ingredients, then herbal constituent data for the herbal medicines, then potential protein gene targets are identified, then target related information is mapped. So this is how the networks are constructed. This is from another paper where the traditional Chinese medicine is used in ischemic stroke kind of a disorder. Then they try to identify the molecules and the interactions, they try to map, and then they created the network for such a formulation. So if you see, another authors have made traditional Chinese medicine with the 6 different ingredients, 6 different plants. With these 6 plants, they've constructed the pathways and to understand the gene and protein interactions on particular disease. So the network looks very complex, and this is what actual biological system is. It is not simple system where you get one target and one molecule and it goes and acts on that. It is a complex system where every molecule in the plant or in the medicine acts on different targets and elicit the pharmacological response. So this is another paper where the authors have used 4 different plants. They try to map the metabolites from these 4 plants and then categorize them based on the chemistry, and based on the structure activity relationships they identified the targets and then given a proper indication and proper validation for the traditional use. If you see the advantages of this network pharmacology based approaches, the scientific evidence is fully understand the rationale of the traditional formulation. So far we were using traditional formulations without much understanding. Now we can understand the mechanism of action of this traditional medicine, why this combination was made, why not only one plant, why several plants were used, and what indication was given, and all that we can validate. So network-based designing and prescribing of the plant formulations is possible. Then analysis of multiple bioactives, studying synergism between these bioactives, as I told you. Then botanical biomarkers for quality control. So what marker to be identified and analyzed for quality control purposes, that also we can validate using this network-based pharmacology-based approaches. Another thing in the modern pharmacology from the plant-based medicines, it can help in develop new leads. We can identify some new molecules from the natural products and we can develop a phytopharmaceutical drug kind of a product, understand the mechanism of action of the drugs, determine the possible side effects of the drugs. That is also possible using this approach. Then predicting new indications, then predicting possible drug-drug interactions, and we can also repurpose some of the drugs using this approach. We can also propose a new purpose or new positioning for a drug. So another aspect is in the drug research as such. In the drug research, we can use this network pharmacology for identifying novel drug targets. What are the new genes or new proteins? How we can identify those targets and reduce the cost and time for using in silico approaches. Understanding signaling pathway of disease types. So this is very important, how the signaling pathway is happening in different diseases. When there is an imbalance, how the molecules, how the genes and proteins react. And therapeutics of multi-gene dependent diseases also possible. Then discovery of disease causing genes is also possible. Then having these wider applications, this network pharmacology is becoming very, very popular. So this is one of the things I wanted to share so that researchers can use these kind of modern approaches in the traditional medicine, and they can discover new ingredients or new formulations for the benefit of the mankind. So I just want to take these 2 or 3 slides. There are a lot of new technologies evolving in the extraction methodologies also. Conventionally, we used to use maceration, oxidation, and decoction, these kind of techniques. But now there are a lot of advances happening, like ultrasound assisted extraction, microwave assisted extraction, pressurized liquid extraction, supercritical extraction, pulse electric field extraction, enzyme assisted extraction. These are more newer technologies, more eco-friendly, more greener technologies, using less solvents or using only water as a solvent, and use the potential of these technologies for the conversion of plant material to the extract. So if you see different type of extractions are given in this picture. So starting from our conventional techniques, so we can talk about microwave assisted extraction, enzyme assisted extraction, or ultrasound assisted extraction, or supercritical fluid extraction. All these technologies are being employed for the extraction and isolation of different constituents from the herbal ingredients or herbal medicines. So this is one of the advances. Probably researchers who are interested in can take up these kind of greener technologies also. These greener technologies help in better isolation of the active constituents. It reduces the energy consumption. It uses alternative solvents other than the toxic solvents, renewable natural products, high quality of extracts, more selective extraction can be done, more safer products, and more less environmental impact products. So these are very, very important in the research of natural products. So the network pharmacology as well as the green extraction technology, these two things I wanted to cover so that it can benefit for the audience of this lecture. Thank you very much. And again, I thank you for giving me this opportunity.
Thank you, sir. We'll move on with the next presentation. Dr. Sundaram, there is a question for you, sir, in the meeting chat. Which method is better for HPTLC or HPLC for herbals?
Okay.
All markers are not active yet. Use it. If we are using it, how like that they have asked. I hope you can see the questions in the meeting chat.
Yeah. I have seen. Yeah.
Yes.
Sure.
Kindly answer that question, Arun, sir.
Yeah. See, HPTLC is very, very appropriate use as a fingerprinting technique. So as we spoke also, plants contain several molecules and it may be not possible to profile all the molecules for the quality control purposes. So when we do a HPTLC profiling, we get the holistic nature of the herb. So if there are seasonal variation, there are geographical variations, and many times there is an intentional adulteration also happens. So such intentional adulterations or substitution happens in the industry. So to just rule out all those quality issues, we can use HPTLC for the profiling. Whereas HPLC is used for the quantitative estimation of the marker compounds. Suppose, for example, you say you are having a ashwagandha root. Ashwagandha root, you can just do the quality control by HPTLC, but the assay part of it you have to do by HPLC to understand the quantum of the vithanolides present in that. That is very, very important. Sometimes there are some negative markers also. Some negative markers should be within a percentage. Say vithaferin A should be less than 0.1%. So that also can be effectively controlled using HPLC method only. I hope I have clarified this question. And yes, this AI and ML definitely helps in the herbal research. So few years before, we thought these techniques are not going to be helpful for the biological research. But definitely they are going to be helpful because, as I told, this network pharmacology itself is all AI based and ML based techniques, and it gives a lot of, because once we have the database of the genes and the database of the proteins and the database of the molecules in your hair, then you can reconstruct a lot of models and identify newer therapeutic applications for the traditional medicines, and then position them differently in a market, and you can use even commercial benefit out of it. Definitely it is going to be helping. Thank you so much.
Thank you so much, sir. Thank you for clarifying the doubts. And the session was also awesome, sir. Thank you so much.
Thank you.
We'll move on to the next presenter. The keynote speaker of the day will be Mr. Angit Sharma. He is the nursing manager in Sun Pharmaceutical Industries Limited. He is having more than 11 years of experience in analytical department on method development and validation activities. He has graduated from IMS Engineering College with the degree of biotechnology in 2011 and masters in chemistry in 2015. And currently he is pursuing PhD from the University of Transdisciplinary Health Sciences Technology, Bangalore, India. Over to you, sir, for the keynote presentation.
Thank you so much. And I am very honored that you give me the opportunity to, as a keynote speaker in the 10th World Conference on Pharmaceutical Science and Drug Manufacturing. So in pharmaceutical industry, there is a main challenge about the genotoxic impurities. So today I am taking that topic, like identification and quantification of genotoxic impurity. and drug products. So today I'm going to present on that. So I'm audible.
Yes, sir, you are audible, sir. Make sure that your volume is little bit high. It is too low, sir.
Okay, okay. Uh, Sushmita, my screen is visible.
Yes, sir, your home screen, yes, it is visible. Can you just place it in the full screen?
Yeah, sure. So today I'm going to present identification and quantification analysis of genotoxic impurities in pharmaceutical products. So genotoxic impurities can be broadly defined as those impurities that had been demonstrated to cause harmful changes in genotoxic material, regardless of their mechanism. Globally, people suffer from the various health complications due to genotoxic impurities. Recent recommendations from the EU and US FDA that the management of genotoxic impurities and possible genotoxic contaminants in pharmaceutical ingredients at ppm level. So, what are the impurities? Component of the new drug substances, which is not the chemical entity defined as the new drug substance, like unwanted chemicals. Those have no therapeutic value, potentially harmful. Some levels are unavoidable. Like there is a one case study. Roche in 1998 he launched the drug for the HIV. After some time, there is a complaint regarding the strange odor associated with the bottles. When they investigate that, then it results to the ethyl methane sulfonate is a genotoxic impurity is very harmful. So due to this, Roche facing a loss of billions of dollars. So what are genotoxic impurities? Compounds having potential genotoxic impurities and like UV and ionizing radiation, protease inhibitors, nucleoside analog, protein synthesis inhibitors. What is genotoxicity, mutagenicity, and cancer? A broad term that refers to the any of their mechanism by which the changes damage at the both DNA level and chromosomal threshold. Chromosomal damage and mutagenicity agent with specific genes resulting is and cancer is the body is called cancer. So somehow genotoxicity is also induces in the cancer because it affects our genes and chromosome and it also causes somehow it any abnormal growth in the body and it causes the cancer. So, what is the regulatory position for mutagenic impurities in pharmaceutical? Like in 2007, European Medicine Agency Committee give guidelines on the limits of genotoxic impurity. In 2008, FDA drafted guidance for the genotoxic impurities. In 2008, also EMEA Q&A also gives the guidance. And 6 February 2013, ICS gives a guideline M7 for the like the what are the limits for the genotoxic impurity, how to control. And in June 2014, ICS M7 assessment and control of DNA reactive impurities in pharmaceutical to limit potential carcinogenic risks. In 2007, there is a one more amendment to the like year to year there are the like we so many genetic impurities are discovered in recent years. Like in last, there is a impurity discovered by the China, nitrosamine impurity discovered by various sartans. So now it is mandatory to control whenever there is a risk for the nitrosamine impurity, then you have to control it as a at intermediate stage or at main API stage. So in November 2018, there is a development of the list of compound in the second amendment and alternate calculation for compound specific limits. In June 2020, ICH M7 Q&A, it's a document available now on the ICH website. Genotoxic impurities structure alerts. A classification of structure features commonly associated with mutagenicity. Like there is a how we know there is a chances of genotoxic impurity. Like there are some structure alerts in the like there is a structure of the compound. So we can say on the basis of structure there is a chances of that kind of genotoxic impurity formation. So we have to control that genotoxic impurity at their specific level. Like aldehydes and their nitro compounds like hydrogens and nitrosamine, azo compounds, carbamates. So there are the like allyl halide. There are the structure alerts on the basis of like compound having the nitro group or hydrogens. So there are the we have to control. We have to check the what are the possible genotoxic impurities are there and how and we develop the method to control that. So what are the genotoxic impurities classification? There are 5 types of genotoxic impurities like class 1, 2, 3, 4, 5. In class 1, impurities are known to be both genotoxic and carcinogenic. And in class 2, impurities are genotoxic but with unknown carcinogenic potential. In class 3, impurities are structure alerts with unknown genotoxic potential and for which the structures have an altering structure related to the API or contain an altering function group moiety that is shared with the structure of the API. In class 5, impurities are not structure alerts, thus controlled as ordinary impurity covered by the ICH Q3 guideline. Like there is a according to the ICH M7, there is a acceptable intake or permissible daily exposure limit. Like for the benzyl chloride, there is a daily limit is 41 microgram per day, and these are the limits for the so many compounds. Sources of impurities in pharmaceutical products, like there are from starting material to the main API, like there is a starting material, then there are so many intermediate stages and there are so many byproducts for that, and so many reagents we are using in the formation of a API, and so many solvents and catalysts are also there, and some degradation product are also forming in whole process of the API formation. Like any small molecule API will always carry with it process control material impurities, and this could be a safety concern, specifically if their toxicity could not be monitored in humans, if they were mutagenic carcinogenic. Like in for we can say starting material, there are so many chances of nitroso and hydrogen. In intermediate stage, we are also there is a chances of nit. So wherever the secondary amine is or secondary or tertiary amine is present in the structure of a compound, and it takes the nitrate from the water, SCl, or from the atmosphere. Nitrate is present is everywhere, and it forms the nitrosamine impurity. That's why nitrosamine impurity is very harmful. Need for the study. Genotoxicity always have become an integral component of a regulatory requirement. Compounds which are positive in these states have the potential to be human carcinogenic or mutagens, so it used in prediction. Like every country having their own regulatory guidelines, like Europe having EU, US having a FDA, like all the countries having their regulatory body for the, and they all gives according to their requirement, they give the limited limits for the specific genotoxic impurities. The methods and action points, like raw material reagents, intermediate impurities are evaluated for genotoxic control through literature, or there are so many softwares like TopCat, Derek, Case Ultra, Multicase. So this prediction based on the static based, or there is a alert expert rule based, or secondary method should be statics. So there are the two methods, like previously we have identified these kind of impurities are present in these kind compounds are present in the structure of the intermediates or API. So these are the possible impurities can be formed. So if potential genotoxic impurity, then control an API with permissible limit. If PGI observed about level, then MA stats should be published. These software are the like in vitro test, we can only predict, and MS test is confirmed. Like if MS test is negative, then it is normal impurity. If MS test is positive, so then we have to control impurity with its permissible limit. Threshold TTC level calculation of the any impurity, like our how we can calculate, like there is a raw material A is used during synthesis of an API, is classified as possible carcinogen to human. Maximum daily dose of the API is 0.375 gram, and so how we can calculate limit. So our TTC is 1.5 microgram per day. Dose is equal to 0.375 gram per day. So then we divide the TTC upon the dose. So we can quantify the genotoxic impurities by the so many techniques like present in the analytical department. Like main is LCMS, because its level is very low, like in 0.7 ppm, 1 ppm.
Sir? Sir, can you hear me? Ankit, sir? Sorry, sir, we can't hear you. So can we conclude the presentation? I hope he is facing some issue. Mr. Ankit? Okay, then we'll kindly wind up with the presentation of his. I hope he was at the end of his presentation. Thank you, keynote speakers, for your valuable contribution for day 2 of the Pharma conference. I hope all the topics that have been dealt was really useful for the participants who have joined. We'll move within 5 minutes. We'll move on with the next presentations, that is scientific paper presentations. I hope that. Sorry. I hope there is two presentations. First hall will be happening in the main hall, and the panel session will be you'll be joining with the rooms. The breakout rooms have been opened. The presenters who are there in the main hall can stay in the main hall, and the presenters in the panel session hall A can join in the breakout room. I repeat, the session, the scientific paper presentations who are doing in the main hall can stay in the main hall itself. And the other presentations, that is 10 presentations from the parallel session hall A, can join the breakout room. I'd like to guide to the breakout room. If you are joined in the laptop, in the more session, you can see the breakout rooms at the down column. In that, you can click the breakout room hall A and you can join. In the mobile, it will be seen in the left hand corner, upper corner, there will be breakout room. You can click over that and you can join the hall A. I hope I am clear. I'd like to call out the names who have to join the breakout room. Guram Naga Aishwarya, Ravindra Babu, Shiva Ranjani, Deepti K. Prabhakar, Maitri Sunil Kumar, Manisha, Moitar G. Devi, Ravindran A.P., Dr. Nandini M.S., and Venuka Devi. The 10 participants from the Hall A presentations can join Hall A respectively.
Hello.
Yes, ma'am.
I think you made a call for Manisha. Which Manisha did you call out?
Sorry.
I think you called out the name Manisha. I wonder why, whether you called out my
Yes,
name
ma'am.
or.
You have to join the breakout room.
Okay, just
Your
a minute.
session will be going on in the breakout room.
Oh,
Kindly
just a
join.
minute. So which one? Which breakout room shall I join?
Manisha, is it Manisha Rajput?
No, no, no.
Raj Purohit?
No. I'm not Manisha Rajpurohit. I'm Dr.
Okay,
Manisha.
ma'am. Sorry, the Manisha Raj Purohit from the Car Kelly College of Pharmacy only is joining, has to join the breakout room.
Okay, that's fine. Thank you.
Yes, ma'am. Thank you. Your Manisha Nitin.
No, I'm Manisha Shukla.
You have the presentation today, ma'am?
No, it was yesterday. It's done.
Okay, fine. That will not be a problem, ma'am. You can join any session, whichever you want to witness.
Okay, thank you.
Thank you. We'll be starting the session in 5 minutes. I'd like to welcome the session chair of the two sessions, Dr. K. Lakshmi, Professor and Dean, Chettinad School of Pharmaceutical Sciences, Dr. S. M. Vanmati, Assistant Professor, Sri Venkateshwara College of Pharmacy, Ariyur, Puducherry, and the session chair of the Hall A, Dr. Padma M. Parkash, Principal, Oxford College of Pharmacy. We welcome you, dear evaluators, for the sessions. Okay, we'll be starting the session in a minute. I hope all the participants are there in the parallel session A. I repeat, the sessions, the 10 presentations that is in the parallel session Hall A will be happening in the breakout room which is named as Hall A. None of the presentations of theirs will be happening in the main hall. I'd like to again welcome Dr. K. Lakshmi, Professor and Dean, Chettinad School of Pharmaceutical Sciences, and Dr. S. M. Vanmati, Professor, Sri Venkateshwara College of Pharmacy, Ariyur, Puducherry. Welcome you evaluators. Am I audible?
Yeah, it's audible. Thank you.
Yes, ma'am. I hope you have your evaluation performance. I would like to dictate the rules for the scientific session.
Background andre nin PPT
Those who are not presenting, kindly make yourself in a mute mode.
ನನಗೆ ಅದು ಗೊತ್ತಿತ್ತು. ನಾವೆಲ್ಲ ಕೊಡ್ತೀವಿ ಹಂಗೆ. ವರ್ಚುವಲ್ ಬ್ಯಾಕ್ಗ್ರೌಂಡ್ ನ ಡೌನ್ಲೋಡ್ ಮಾಡಿ ಇಟ್ಕೋಬೇಕಂತ ಕಾಣುತ್ತೆ. ಇಲ್ಲಿ ವರ್ಚುವಲ್ ಬ್ಯಾಕ್ಗ್ರೌಂಡ್ ನ ನೀನ್ ಕ್ಲಿಕ್ ಮಾಡಿದ್ರೆ ಬರ್ತದೆ ಅದು.
I repeat, those who are not presenting and other than the session chairs, kindly make yourself in the mute mode. There will be 5 minutes given for the poster presentation and 10 minutes will be given for the oral presentation. We will be notifying you once the 9 minutes is over. The 1 minute balance will be taken for the question sessions from the session chairs. Please don't mistake us if we interrupt the session once the time is over. The set of session chairs will be evaluating the sessions based on the criteria given. So make sure the time is being managed properly, and make sure that you are ready with the PPT presentations. You should be able to make it properly, telecasting to the live stream Zoom. I'd like to call our first presenter of the session, R. Vimala Vadini from MTPG and RIHS for presenting her paper.
Good morning, ma'am. Am I audible?
Yes, you are audible, but your video, we can see only your head. Sorry, can you just move upward or something like that? Yes. Yes, now it is fine, ma'am.
Okay. Can I share the screen?
Yes, you can. Session chairs, you are open to ask any questions to the presenters at the end of each session. You can unmute yourself and you can speak. Ma'am, can you start? Vimala Vadini? Ma'am? Ma'am, are you facing some issue? Can we move on with the next presenter? Sorry, we can't hear you. Okay, we'll move on with the... Okay, I hope she's not in the Zoom. We'll move on with the next presenter, Dr. Lata Kothpalli from Dr. D.Y. Patel Institute of Pharmaceutical Sciences for presenting her e-oral presentation. Dr. Lata Kothpalli.
Can you see, ma'am?
Ma'am, your voice is breaking.
Can you see this, ma'am? My screen?
No, we can't.
Hello, ma'am.
Yes, ma'am, we can't see your screen. We can see your video only.
Okay.
We'll wait or we'll move on with the next presenter, ma'am.
Yeah, it is, screen is loading and showing.
I hope there are some internet issues. Make sure that you are sorting out with that, ma'am. Dr. Lata Kothapalli is here. Okay, we'll move on with the next presenter, Dr. Preeti G. Karade.
Yes, ma'am, I'm here.
Yes, Dr. Preeti, you can go.
Okay.
She will be presenting her e-oral presentation. Kindly note session shares.
Madam, someone is sharing the screen, so I can read the message. You cannot start the screen.
Yes, ma'am. Now you can. I hope now you can share your screen. It has been stopped from Ms. Vimala Vatini. Kindly share it now. Yes.
Ma'am, is it visible?
Yes, it is visible. Kindly make it in a full screen mode.
Okay. Is it okay now?
Yes, ma'am. Now it is perfect. You can start.
Okay. Good morning, one and all here. Before I start, I would like to thank you, organizers and Association of Pharmaceutical Research, for giving me this opportunity to present my work. My work it is related to design and evaluation of microsponge for colon-targeted drug delivery of secnidazole. Amoebiasis. Amoebiasis, it is a common infection of human gastrointestinal tract. It is closely related to poor sanitation and socio-economic status. It can be treated with different types of nitroimidazoles. Secnidazole, this is new version of nitroimidazole. It can be used in the colonic infection management, but its traditional oral dosage form, it is associated with certain side effects and degradation of drug in the gastrointestinal tract. So we have decided to use the microsponge. The microsponge, they are spherical, they are having good flow property, better compression property, and they are provided, and because of their smaller structure, as they are smaller in size, they can be effectively taken up by the macrophages which are present in the colon. So the aim of my work was to design a new colon-specific drug delivery system containing secnidazole microsponge. The two different approaches that were explored. In the first part, the core tablet were prepared, and in the second part, the core tablets they were coated with different composition of xanthan gum and phytosan. So my formulation part, it is divided under 3 parts. In the first part, drug-loaded microsponge were prepared. In the second part, 4 tablet formulation containing drug-loaded microsponge was prepared. And in the third part, the core tablet, it was coated with different compositions of xanthan gum and chitosan. So this is first part, preparation of microsponge. So for the preparation of microsponge, different compositions they were used. The table shows the presence of different composition, and the microsponge they were prepared by quasi-emulsion solvent diffusion method. For that, internal phase and external phase they were separately prepared. Drug itself, Utrazit RS100 and organic solvent as dichloromethane. It was used in the internal phase. Internal phase was then added slowly with continuous stirring to the external phase, and the microsponge they were separated by filtration. Here we are used 2 independent variables, use of dichloromethane and Utrazit S100. They were used as independent variable, and its effect on different parameters were checked. On that basis, one of the batch, either MS1 for microsponge 1, 2, 3, and 4. So one batch was optimized, and that batch, it was used for the formulation of core tablet. This is the composition for the formulation of core tablet. Microsponge drug-loaded microsponge, they were used. Sodium starch glycolate, microcrystalline cellulose, magnesium stearate, talc, they were mixed properly, and the mixture was directly compressed using a flat punch of 6 mm on a 10-station rotary tablet punching machine. Now, the core tablet which was prepared, it was coated for colon targeting. It was coated with different compositions of chitosan and xanthan gum. CCT1 stands for compression coated tablet formulation 1, CCT2 for formulation 2, and for formulation 3. So we have used 3 different, we have formulated 3 different formulations. Evaluation part, evaluation of secnidazole microsponge, it was carried out with the help of different characteristics, different evaluation parameters, like particle size analysis was carried out, entrapment efficiency was determined, in vitro drug release, scanning electron microscopy, differential scanning colorimetry was used to determine interaction of the drug and formulation. Evaluation of core tablet was done with the help of post-compression characteristics and disintegration test and dissolution test in the absence of radical content. While colon-targeted compression-coated tablet all the post-compression characteristics were determined, as well as the drug release study was carried out in the presence of radical content. So pre-formulation, the melting point was found to be 75. IR spectra indicate that there is no interaction between the drug and excipients. It was again confirmed with the help of differential scanning thermogram, where the drug, it has a sharp melting point, while the formulation does not, in case of formulation, the melting point was, there is no sharp melting point. So this is about evaluation of microsponge, percentage yield, entrapment efficiency, and percentage drug release. It was found maximum for the formulation of core tablet. These are the results which are obtained: 3D response surface and contour plot, which are obtained by design statistics software, where left upper and lower two graphs, they indicate the effect of polymer as well as dichloromethane on entrapment efficiency and drug release, while the right two parts, they show its effect on the drug release. So as the volume of dichloromethane increases, it was found that the solubilization of drug, it was also increased in the organic solvent, and entrapment efficiency was increased. While in case of drug release, in vitro drug release, as the concentration of polymer was increased, the drug release was found to be decreased. This is related to compression coated formulations, where thickness, hardness, friability, weight variation, and drug release was determined. And we have found that compression tablet formulation 1, they show maximum drug release, and that was found to be 97.53%. It is in the presence of radical content and in the absence of radical content. So here, in the presence of radical content, the drug release was found to be maximum. These are scanning electron microphotographs which are obtained in the first. This is related to plain microsponge, where all the microsponge, they were spherical in shape. This the second microphotograph indicates microsponge. Am I audible, ma'am? And in the third, it shows
yes, ma'am, you are audible.
And the broken tablet, SEM of broken tablet, it was taken. So conclusion, it is colon targeted tablet formulation, including microsponge. It was prepared based on the approach of triggering mechanism of microflora activation, which gives delivery of the drug to the colon with minimum release in the stomach and small intestine. These are some of the references which I have used. Thank you, ma'am.
Thank you so much, ma'am. Session chairs, any questions?
Yes. Ma'am, did you compare with the market available formulation?
Yes, we have compared with the marketed formulation, but the study was carried out only up to 12 hours. And for the microsponge and colon targeted drug delivery system, we have conducted the study up to 24 hours. That's why I have not included in the slide.
Okay, which tool you have used for all this surface response tools?
Sorry, madam?
Which tool you have used for statistical analysis?
For statistical analysis, madam, design expert software, Statis, was used.
Okay. Okay.
Thank you, ma'am.
Thank you, ma'am. We'll move on with the next presenter. R. Vimala Vadani, are you ready?
Yes, ma'am.
Yes. She'll be doing the e-poster presentation. Over to you, ma'am.
Thank you, ma'am. Can you see the slide, ma'am?
Yes, ma'am, we can see your slide. Kindly make it as full screen.
Can you see it, ma'am?
Full screen, ma'am. Can you make it as a full screen?
Yeah, I'll do.
Yes, you can start your presentation now.
Yes. Good morning, everyone. This is an in silico screening of NMV, that is Neeradi Muthuvaladi capsule, a Siddha formulation for its anti-inflammatory and anti-asthmatic activity. It is a drug repurposing study. So we know that asthma is a chronic disease and it involves many immunological and inflammatory molecular proteins, of which CD4, Janus kinase, JAK-STAT, phospholipase A2, COX, LOX, histamine, prostaglandins are involved, implicated in this. And NMB is a Siddha medicine, which is currently used for anti-cancer and anti-psoriasis drugs for a long time. And this GCMS evaluation, we found out that it had nearly 50 chemical constituents, of which aziridin, deoxy, spergolin, piperazine, mannitol, cytothionin, citrulline, some of the components exhibited anti-asthmatic activity, specifically anti-asthmatic activity. And from this, we took some 5 ligands, that is arginine, aziridin, APDP, an aziridin derivative, and another derivative AMB, and citrulline, and we docked it with these proteins, molecular proteins. So it was a drug repurposing study. It is used only as an anti-cancer and anti-psoriasis drug. From the GCMS report, we saw it is having some anti-asthmatic constituents also. So we wanted to find out, is it effective in anti-asthmatic? So whatever the molecular proteins involved in the asthmatic pathway pertaining to inflammation and immunology, we did the docking and we have the reports. So for this, the structure of the ligands, the 5 ligands was imported and drawn in Marvin sketch. And this was docked with molecular proteins that were imported from the RCSB protein data bank, and the in silico docking studies were done. This was the table, that is the docking score of the molecular proteins with the 9 molecular proteins, that is CD4, JAK, STAT, PLA, COX. PLA2 is phospholipase A2, COX, LOX, PGD2, and leukotriene 4 hydroxylase, and histamine 1 receptor. And the study showed it gave a very good binding energy below -5. Below -5 is acceptable, and for some ligands, the values were even -8. Like for example, with prostaglandin D2, arginine and citrulline gave even less than -8 or -7 and even -9. And it gave good hydrogen bonds, which implicates the drug is having good interaction with these proteins, anti-asthmatic activity. So to conclude, the anti-asthmatic and anti-inflammatory activity of NME may be due to its propensity to modulate immune, allergic, and inflammatory pathways of asthma. And this study recommends futuristic in vivo and in vitro studies of NMB to establish its anti-asthmatic activity in allergy and inflammatory asthma. These are the references. Thank you.
Okay, madam, as you mentioned, the different ligands and the different proteins you have chosen, right?
Yes, ma'am. Yes, ma'am.
That's what you could have given a comparison, like the ligand with the different proteins.
Yeah, that's
in
what
table
I have
you
given.
have. That's what
Yeah.
in table you have given, but some kind of graphical representation with the effect of ligand with the different proteins will have better understanding.
Yeah, but
we have to scroll through that, then we have to find out what the value is.
Yes, ma'am. Actually, I prepared, but the constraint of space was one reason. And this is only a part of what we have done. Actually, we took 13 ligands and with all these proteins. So I couldn't put all the details, only important, whichever had good work. Even others have nearly, there are some 45 docking scores. So whatever was the best, I tried to put in the given space. And
Okay, but the chemical constituents, whatever were listed, are from the literature, right? You have not done the GCM.
No, we actually gave GCMS and we got 50 chemical constituents. And these 5 or 7 I have put in the introduction part. Those alone are given in the literature and they showed anti-asthmatic activity. This drug is used as an anti-cancer drug. It is a drug repurposing study. We are telling that it can be used as anti-asthmatic in allergic or inflammatory asthma conditions.
Okay. Okay.
Ma'am, can I go to the next presentation?
Yes, ma'am.
Yes, ma'am. We can move on with the next presentation. That is also poster presentation.
नहीं सर ऑफिस का नंबर है
बजाज का नंबर है तो रजिस्टर नहीं
आई थिंक इससे भी चल रहा है
नहीं इससे नहीं चल रहा है
एक नंबर है 9580
9588032
67
67
Ma'am. Can I?
The poster is not seen.
Next.
No, ma'am, your screen is not visible. In a sense,
No,
your screen is there in the other document or something like that.
No, ma'am.
Yes, ma'am. Kindly make it out. Okay, fine. Yes, ma'am. It is visible.
Okay, the next presentation is the computational docking studies of pionidine with molecular proteins of PI3K, AKT, and mTOR pathway. So this pathway is involved in the pathophysiology of various metabolic inflammation, apoptosis, and oxidative stress, of which the main proteins are phosphoinositol 3 kinase, protein kinase B, that is AKT, mTOR, PDK, that is pyruvate dehydrogenase kinase, and FKHR, fork head box protein not 1, and BCL2. And operation of this signaling cascade has been implicated in the lifestyle disorders, and molecules which can moderate this pathway can have significant therapeutic potential. And pionidine is a flavonoid of the anthocyanin group. It is found broadly in the berries and the cherries. It gives a purple color to the plant, to the fruits. It has potent antioxidant, anti-apoptotic, and anti-cancer properties. And literature shows that this pionidine has multiple hydroxyl groups in its structure. So we propose that presence of multiple hydroxyl groups can modulate the molecular proteins of this PI3K, AKT, and mTOR pathway. Hence, the aim of our study was to determine the in silico interaction of pionidine with 6 molecular proteins of this pathway, namely PI3K, AKT, mTOR, PDK, FKHR, and BCL2 using Autodoc software. Coming to the materials and methods, the structure of the ligand pionidine was imported from PubChem and drawn in Marvin sketch, and the molecular proteins were imported from Protein Data Bank, and the in silico docking studies were done using Autodoc 4.2 software. The table 1 gives the docking score. In this, we can see that PI3K has the maximum score, that is minus 8.08. Next is PDK2, it is minus 7.53. And rest all had minus 5, which is acceptable, except mTOR. mTOR showed only minus 4.8, which is not acceptable binding energy. And it also had sufficient hydrogen bonds. And these were the docking images of pionidine. The first one is PI3K with pionidine. The next is AKT with pionidine. And the last one is FKHR with pionidine. So I have taken the images of which gave a very good binding energy, that is -7, -8, like that. And to conclude, the inference of our study shows that pionidine shows profound interaction with 5 proteins of this pathway. And pionidine is a flavonoid. It has a flavonucleus with ring C and 2 aromatic rings, that is A and B. Studies show the presence of these rings along with phenoxy radicals and the multiple hydroxyl group of B ring of pionidine causes electron delocalization and hydrogen bonding ability, which can provide antioxidant and other activities, free radical scavenging activities, which can contribute to this pathway modulation activity. Also, since of C2-C3 double bond structure of flavonoid can give potent PI3K inhibitory activity. So maybe that is the reason we are getting very good energy score for PI3K docking, which was nearly minus 8.08. So to conclude, pionidine can moderate this pathway, and hence it has significant therapeutic potential in many lifestyle diseases. Thank you for the opportunity.
Okay, madam, if I have to check whether this particular moiety can cross the blood-brain barrier, what should I do?
You should
Using
go for
this autodoc. If I have to check whether this moiety
no, you can go for different software. You have ADMET for pharmacokinetic studies. You have ADMET, ma'am. For toxicological studies, you have Osiris software. So you have different softwares.
Okay, so we cannot do with this.
No, actually, I haven't tried. We tried that ADMET pharmacokinetics studies.
Okay. Okay, thank you.
Thank you, ma'am.
Yes, ma'am. Thank you so much.
Thank you, ma'am.
We'll move on with the next presenter. Dr. Lata Kotapalli is there. Dr. Lata? I hope she's not there. We'll move on with the next presentation. Mycela A. D. Alva from University of Santo Tomas, Philippines.
Hello, good morning.
Yes, good morning.
Yes.
You can carry on with your presentation.
Yeah, I'll just start sharing my presentation.
Have you started?
Sharing.
Yes, yes, we can. Yes, it is visible. You can start with your presentation.
Okay, so I'm gonna start now, okay? A pleasant day to the distinguished organizers and participants of this conference. Okay, on behalf of my co-authors, Mr. Charlie Cruz and Dr. Ross Vasquez, I am Micaela A. de Alva from the University of Santo Tomas, Philippines, and I am here to present our study entitled The Neuroprotective Effect of Hydroalcoholic Extract from Sargassum silicosum C. agar on ethanol-induced dementia in Sprague-Dawley rats. So first define dementia by the WHO. Dementia is a syndrome that can be caused by several diseases which over time destroy nerve cells and damage the brain, and usually leading to deterioration in cognitive functions. So it also affects each person in a different way, but the common symptoms are the following: memory loss. Second is the difficulty in performing familiar tasks. Third is problems with language. And lastly, changes in personality. WHO also recognizes dementia as a public health priority, and as described by the Alzheimer's Disease International, someone in the world develops dementia in every 3 seconds. And in just 2019, there are over 55 million people worldwide living with dementia, and this number may double every 20 years, reaching 78 million in 2030 and 139 million in 2050. And in the Philippines, there were already 3,368 deaths in a span of 6 years in 2012 to 2018. And in just 1 year, in the year of 2002, there are recorded deaths of 2,010 deaths. And from the Philippine Statistics Authority, the prevalence of dementia in 2050 will be 1,159,000 rather. Okay. Dementia is also defined as the overall term for a particular group of symptoms. It also has several causes that reflect specific changes in the brain, and these are the following causes of the dementia: Alzheimer's disease, cerebrovascular disease, frontotemporal degeneration, hippocampal sclerosis, Lewy body disease, and Parkinson's diseases. Each common causes have its respective brain changes that affects the dementia symptoms. There is also no cure for dementia, but these are the medications that is used to help manage the dementia symptoms. First is the cholinesterase inhibitors, antipsychotics, antidepressants, and nootropics. Okay. Sargassum polysystem sea agar belongs to the family of the Sargassaceae and generally recognized as gulfweed or sea holly. The genus Sargassum contains approximately 400 species distributed in the tropical and subtropical areas of the world, in which in the Philippines it is also called as aragan, boto-boto, lusay-lusay, and samo. These are also the following studies that already prove the different pharmacological activities of the Sargassum species, such as the wound healing, anti-allergic, antioxidant, and anti-cancer, hypoglycemic and hypolipidemic, anti-inflammatory, and the anti-tumor activity. And in this study, we will evaluate the neuroprotective potential of the hydroalcoholic extract from the Sargassum polysystem on ethanol-induced dementia in Sprague-Dawley rats. And these are the following methods and procedures that are done. First, the sargassum or the seaweeds were collected and identified and was authenticated by the Marine Science Institute of the University of the Philippines. Then it was extracted with 30% ethanol water to extract the hydroalcoholic extract. And it is assumed that the fluorotannins were extracted through this solvent. After which it is subjected to analytical procedures, which is the total phenolic content and the organoleptic analysis. The animals will be also prepared for the acute oral toxicity test following the OECD guidelines 423 with an approved IACUC protocol from the University of Santo Tomas. The different treatments will be also administered before the behavioral test, which are the passive avoidance test, 8-arm radial maze, and the novel object recognition test. After the behavioral test, blood will be collected and will be subjected to acetylcholinesterase activity assay using the Sigma Aldrich MAK 119 kit. Okay, so these are the experimental procedures that were done. These are the following groups for each treatment. Each group has 5 rats each, in which the group 1 received NSS, group 2 received ethanol as the induced dementia. Group 3 will receive the positive control, the piracetam plus ethanol. Group 4, 5, and 6 will receive different concentrations of the sargassum extracts and will be also administered ethanol. Ethanol will be administered on the first day of the behavioral test, unlike of the extracts, the positive control and the normal control will be administered a week before the behavioral test are. Okay, so this is the 8-arm radial maze. Prior to the actual test, rats will be given a training session in which it familiarizes the platform with baited arms. Only 4 arms were baited and constant for all test subjects, and the number of entries to baited and unbaited arms were observed to compute for the percent working memory and percent reference memory. Oops, sorry. Okay, so there are 2 identical compartments in the passive avoidance test, which are the bright and dark compartments. So rat will be placed first in the illuminated compartment and the gate between the compartments will be open. When the rat enters the dark compartment, the gate will close and the rat will be given an electrical foot shock. So the step-through latency time will be compared to the time taken for the rat to enter the dark compartment to the training day versus the test session. Okay. And on the novel object recognition test, there will be 3 different sessions, which will be habituation, familiarization, and the actual test session. The amount of time spent exploring both novel and familiar objects will be recorded. The discrimination index will be analyzed to determine the difference in time spent to explore novel and familiar objects relative to the total time spent exploring both objects. And these are the following results gathered. In the organoleptic analysis, after the crude drug was extracted with 30% ethanol, the lyophilized powder appeared as a yellowish-brown crystalline powder with a faint fishy odor with a percent yield of 3.627. The total phenolic content was estimated using the Follen-Shakal reagent, in which we use the fluoroglycinol as the standard to calculate the phenolic content in milligrams of fluoroglycinol equivalents. The total phenolic content calculated against the standard curve of fluoroglycinol with an R-squared of 0.9953 and has a mean of 41.03 milligrams PGE per gram. And these are the results of the acute oral toxicity test, in which the 2000 milligrams per kilograms is non-toxic, with the rats showing no more lesions in the liver. And in the 8-arm radial maze, or the ER maze, a separate one-way ANOVA test for each variable was used to analyze the percentage errors for working and reference memory statistically. Results showed that among the different concentrations of the extracts, 500 milligrams per kilogram body weight of the HASP has a significant difference to the negative control, with a p-value of the 0.045. In the NOR test, a value near 2 + 1 shows more exploration to the novel object, meaning that the rat could identify or recognize that a novel object replaced the familiar object. So in the group 5, showed a slightly higher value among the concentrations than the group 6. The values of the excess showed a significant difference from the negative control group. The
You
PA
have
test,
only 1 minute left. Make sure that you're wrapping up with your presentation.
yes, using one-way ANOVA test, group 5 was statistically significant among the concentrations, with group 2 having a p-value of 0.003. And in the ACHE, it showed that the increasing concentration of extract has a lower activity, suggesting its neuroprotective activity. And to summarize and to conclude this study, rats treated with a 500 milligrams per kilogram body weight AESP is consistently displayed improved memory and behavioral activity in the different behavioral tests. The HASP treatments also decreased the acetylcholinesterase level in rats' blood comparable to the ethanol-treated control group, and these findings also suggest that the HASP has the potential neuroprotective properties and can be a potential source of the anticholinesterase agents. We therefore recommend to future directions of the study to isolate and identify bioactive compounds present in the HASP, and to conduct more of the chronic toxicity test, other biochemical tests, and histopathological tests, and to confirm the mechanism of action of this HASP by other behavioral tests like the Morris-Water maze and IntelliCage. Okay, and these are the following references I used. Thank you for your kind attention. That's all. Thank you.
Thank you so much. Any questions?
Yes. Okay, so is this, what is the nature of this plant?
The Sargassum polysystem sea otter is a brown seaweed that is endemic, especially to the subtropical and tropical.
Which part of the plant you have used?
We used the whole thallus, or the whole Sargasso plant, in the study.
Okay. So you have done one-way ANOVA, right?
Yes.
Which statistical tool you used?
Yes, pardon us.
Which statistical tool you have used to calculate one-way ANOVA?
We use the SPSS program in computing the one-way ANOVA test.
Okay. So why do you use ANOVA there?
What?
Why do you use ANOVA there? You know the concept of using ANOVA?
Yes, we use the ANOVA because we would like to compare or to look for the significant difference of the concentrations for the negative control group, or within the group.
Yes. Okay. Thank you.
Thank you, Michaela.
Thank you.
We'll move on with the next presenter, Dr. Japal A. Soni from Arihant School of Pharmacy and Bio Research Institute, Gujarat, India, for oral presentation.
Hello. Good morning, ma'am. Am I audible to you?
Yes, ma'am, you are audible.
Okay, ma'am, give me 1 minute to share my screen.
Yes, yes, you can.
Ma'am, is my screen visible to you? Hello.
Yes, ma'am. Yes, yes, it is visible now.
And my presentation?
Yes, ma'am, we can see. Genotoxicity
Okay.
study. Yes, we can see.
Yes, yes. Okay, so myself, Dr. Jalpa Sooni, and I'm going to present on genotoxicity study of pioglitazone and glimepiride combination after 4 weeks in Swiss albino mice. So starting with the presentation, these are some table of contents and introductory part. Basically, this oral hypoglycemic drugs are used globally, and there are 9 different categories of FDA approved medicines which are used in the treatment of diabetes mellitus. Among these 9 categories, metformin, sulfonylureas, and thiazolidines are most studied medicines, and they play the predominant role in the treatment of type 2 diabetes mellitus as per the algorithm advised by the ADA and EASP. Among this, all the medicines which are used in the treatment of type 2 diabetes mellitus, if we study about the sulfonylureas and thiazolidines as a combination, then general combination of available anti-diabetic drugs says as per the advisory committee that insulin sensitizer can be combined with the insulin secretagogue or insulin based agent. So if we study the sulfonylureas and thiazolidine as a combination, they support the strong rationale in type 2 diabetes mellitus, as well as they retard the progression of type 2 diabetes mellitus and decrease the exogenous insulin therapy requirement. And it also offers the sustained glycemic control as well as benefits to reduce the cardiovascular disease risk. However, they have the opposite effect on beta cell functions, as sulfonylureas increase the beta cell function and thiazolidine decrease the beta cell function. Moreover to that, they are also effective in lowering the triglyceride levels and enhancement of HDL levels. But if we consider the risk factor for the detrimental effect on genetic material and DNA of this combination, then hepatic metabolism of the sulfonylurea by the oxidative pathway, as well as possibility of the initiation of bladder cancer by the thiazolidine can be harmful to our DNA and genetic material and can predispose to the genotoxicity. So, from this both the categories, if we choose the pioglitazone from the thiazolidine categories, it improves the glycemic control after long-term use. It is also more beneficial for lipid metabolism than rosiglitazone. And if we choose the glimepiride from the sulfonylureas category, it also provides the improvement in glycemic control, shows better tolerability and safety specificity in a patient with a history of acute myocardial infarction. Uh, Among this antibiotic anti-diabetic drugs are used for the longer period of time in any patient, and it can also affect our DNA as well as genetic material. So genotoxicity study is mandatory for checking the safety of this combination, and this combination can be evaluated for the genotoxicity by the use of different testing procedures as per different regulatory authorities like EU, OECD, and ICH. Moreover to that, WHO scientific groups stress the need to reevaluate the mutagenicity and anti-mutagenicity of timely tested drugs and report even the negative results are obtained. So based on the consideration of this literature review and results of genotoxicity testing of this combination, and to get more insight into the genotoxic potential of the combination, 3 different in vivo testings are used in this present study, that is a mammalian bone marrow chromosomal aberration assay, mammalian bone marrow erythrocytic assay, and sperm abnormality assay to assess the genotoxicity at 3 different levels, that is chromosomal level, cellular level, and germline level. Moreover to that, from this study, we can give the precise results regarding pharmacokinetic parameters and DNA repair mechanisms. Moving to the experimental methodology part, I have used 6 to 8 week old Swiss albino mice and obtained IAEC approval from SSPC Mehsana, Gujarat as per CPCSDA. Experimental protocol of my study was I have included 3 different doses of the combination: lower dose, middle dose, and higher dose, with the inclusion of negative control group and positive control group. In this study, I have evaluated 4 different parameters, that is chromosomal aberration assay, mitotic index, micronucleus formation, and sperm abnormality assay. Uh, I have divided my whole animals into 5 different groups with the use of 5 animal in each group as per OECD guidelines. This is the brief outline of the procedure of all these 3 different assays that I have studied. In case of statistical analysis, each treatment group was compared with the negative control by one-way ANOMA with a significant difference of p-value less than 0.05. Moving to the results of all these assays, first, that is mammalian bone marrow chromosomal assay. So basically, different chromosomal aberrations I have studied, and this figure represent the different kind of chromosomal aberrations like deletion, acentric fragment, gap, exchange, chromatid break, ring formation, chromosomal break, and polyploidy at 100x magnification with the oil immersion lens. And result of this assay denotes that it is divided into 2 parts, that is chromosomal aberration per cell including gaps and chromosomal aberration per cell excluding gaps. So as per the result, we can see that the positive control group shows the significant difference in both the parameters, that is including gap and excluding gap, whereas in all the 3 doses of the combination, only higher dose in excluding gap parameter showed the significant difference of p-value less than 0.05. Moving to the second parameter, that is mitotic index, which is the percentage ratio of number of dividing cell per total number of observed cells, and it shows the cytotoxicity of the combination as well as the cell proliferation rate in bone marrow cells of mice. To study this mitotic index, I have calculated the different phases of cells in cell cycle. Like in the present figure, A represent the normal cell, B1 represent the early prophase, B2 represent the late prophase, C represent the metaphase, and D represent the anaphase. And in case of results, we can see that the positive control group as well as a higher dose of the combination showed the percentage suppression in the mitotic index, and which denotes the cytotoxicity of the present compound. Third parameter was calculated with the use of mammalian bone marrow micronucleus assay. It is again divided into 2 parameters, that is PC to NC ratio as well as extent of micronucleus formation, which can be calculated by observing the polychromatic cell and normochromatic cells. In the present figure, A represent the normochromatic cell and B represent the polychromatic cell, as well as in the right figure, the marked cell contains the micronucleus formation. So suppression in the PC to NC ratio that represent the induction of the cytotoxicity in bone marrow cells of the mice, as well as no in case of results, we can see that the positive control group shows the significant difference in the PC to NC ratio as well as micronucleus formation, whereas in case of the combination, all 3 doses represent the suppression in the PC to NC ratio, and there is no significant changes in the micronucleus formation. A fourth parameter was studied by the use of sperm abnormality assay, that is a representation of the germ cell abnormalities, and it can be calculated by the observation of sperm head abnormalities with a comparison of the normal sperm head. In the present figure, A represent the normal sperm head, B represent the amorphous sperm head, C represent the banana shaped cell, D represent the hookless sperm, and D represent the folded sperm. Moving to the results of this sperm abnormality assay, we can see that the positive control group shows the significant difference in the abnormal cells, as well as in case of the combination high dose shows a significant difference of p value less than 0.01 in the sperm abnormality. So from the above results, if we discuss the present study that in relevance to the genotoxic potential positive control group in which the cyclophosphamide was used, it showed the significant changes in the all parameters that might be attributed to its DNA binding ability and hepatic metabolic activation by microsomal chromosomal P450 mixed function oxidase system. Moreover to that, in case of chromosomal aberrations, that is chromosomal aberration per cell excluding groups, shows the significant changes in higher dose, which represent the clustergenic potential of the combination. Whereas in the above result, we have also seen the structural changes in the chromosomal, which is primary step in the development of the cancer, and it is very complicated process which can be attributed to the either direct DNA damage or replication of DNA damage, or replication of damaged DNA template, or inhibition of DNA synthesis, or it can be also the inhibition of topoisomerase 2. Moreover to that, in the above result, there is
Try to
there
finish up
was a
your
breakage of
presentation.
chromosomal fragment in the presence of acentric fragment, which can predispose to micronucleus formation, as well as in the results, there is also seen that no significant micronucleus formation was observed, although more number of deletion and eccentric fragment was observed. So it can be says that there is a presence of some DNA repair mechanism if we go for the in vivo genotoxicity study, which can be also studied by use of this combination of assays. Other than that, significant suppression in the percentage mitotic index in higher dose, as well as fluctuation in the significance of PC to NC ratio represent the cytotoxic potential of the compound. And it can be said that if any compound has potential to produce somatic cell mutation, they might produce the germ cell mutation, which can be confirmed by the use of sperm abnormality assay. And in the present study, this result also showed the significant abnormality in sperm cells in higher dose. Concluding this present study, this combination have capability to produce structural chromosomal damage, cytotoxicity, and germ cell mutation if it is used at higher dose, but it cannot alter the numerical chromosomal changes if it is given for the 4 weeks. Thank you all for your patience and listening. These are some references of my study. Thank you, everyone.
Okay, Dr. Jalpa, many are consuming this combination tablets. So how to overcome the cytotoxic effect?
Sorry, ma'am, can you repeat the question?
No, actually, many are consuming this combination tablets.
Yes, ma'am.
It's available in the market, right?
Yes, ma'am.
So, how to overcome the genotoxicity or cytotoxicity?
Ma'am, it can be given with either genoprotective agent, or we can decrease the dose, or we can decrease the duration of the treatment with the alternative agents also.
Okay. Okay, thank you.
Thank you, ma'am. We'll move on with the next presenter, Ms. Mariam Bibi from KSP KPGU for her oral presentation. Dr. Jawad?
Yes, ma'am, just 1 second. I'm stopping my screen share.
Yes. Ms. Mariam Bibi?
Yes, ma'am.
Ma'am, we can hear you, ma'am. You can start with your presentation. Try to share your screen.
Is it visible, ma'am?
Not yet. We can see only your mailbox.
Is it visible now?
No, ma'am, your mailbox only we are seeing. You can share and share the window and go to the PPT column.
Okay, thank you.
I hope your PPT is open. Yes, yes, now it is visible.
Okay. Respected jury members and yes, is it visible now?
Kindly put it in the full screen mode. Is it possible?
Yes, ma'am. It is full screen. Is it visible now?
Yes, ma'am, but we can't see your full screen. Kindly share the screen. Are you clicking share screen? I'll just guide you.
Share screen, then entire screen.
Yes, share entire screen.
Okay.
And now go to the PPT column. First share the entire screen.
Yes, I click the window, then I
Yes,
click the
no need of window. Share the third column. Share
entire
entire
screen.
Yes.
But I'm not able to click the share
No,
button.
click over that. Just click over that.
Okay.
Now move on to the PPT.
Now move on to the PPT.
Yes, now we can see
Yes,
you.
now we can see you.
Start with your
Start with
presentation,
the
please.
presentation.
I'm muting
I'm
myself.
muting myself.
You are not
You are
audible.
not audible.
Ma'am, you're not
Ma'am, you are
audible.
not audible.
Unmute yourself,
Unmute
please.
yourself, please.
You have to unmute
You-
yourself.
Is it visible now?
Yes, it is visible. Kindly place it in the full screen mode and start your presentation.
Is it okay now, ma'am?
Yes, yes, it is. You can start.
Okay. Sorry for my inconvenience. Myself, Marie Mandarawala from Krishna School of Pharmacy, Vadodara. Today, the title of my review work is Exploring Precipitation Inhibitors to Improve In Vivo Absorptions of Vitamin D from Supersaturated SNEs. In that, if we see in India, the deficiency of India, mainly the 70% to 100%, the urban and rural, both the area are equally affected. In that, if we see the males are more affected than the female. And India is primarily affected by the problem of rickets due to this vitamin D deficiency. And mainly the cities which have a low sunlight are mostly affected by this deficiency. If we see this chart, the 68.50% peoples are mostly affected by this vitamin D deficiency in India. Here are some of the symptoms which shows the vitamin D deficiency, like the high blood pressure, mood swings, overweight, excessive sweating, fatigue, digestive problems, joint strain. Now, if you see in detail about the vitamin D benefits that control more than the 700 genes in our body, that controls several types of cancers, that strengthen our immune system,
that boost our mood, that affects on weight loss, lower the risk of rheumatoid arthritis, that lower the risk of type 2 diabetes, that can help the blood pressure lower, and also reduce the risk of heart disease. Now here the reference values of 25 OSD for evaluation of vitamin D status in our body. If we have a equal or less than 10 nanogram per ml of serum OHD level, then it called a deficiency of vitamin D. Now the recommended intake of vitamin D, mostly we need a minimum of for elder patients, high level of intake like 800 international units are difficult to achieve. For this reason, we need a supplementation or fortified food or pharmaceutical products of vitamin D as per this chart. But still, we have sources of vitamin D, but some of the causes, like if the lifestyle are indoor type, poor diet, aging, obesity, malabsorption, or a specific type of deficiency, like if we have some kidney disease, chronic liver disease, Crohn's disease, celiac disease, or gastric bypass surgery. At a time, we need a vitamin D supplementation. But with this supplementation, it has been reported that deficiency of absorption of vitamin D by this oral route is approximately 50% because the vitamin D is fat-soluble vitamin. So here the another proof like literature review existing for this innovation technology to address this related problem. Like if we formulate a liposome with this vitamin D, but with liposome, there's some of the disadvantages, like the production cost is very much of high, oxidation and isomerization during the processing and storage. If we prepare a solid lipid nanoparticles, but that have the low drug loading efficiency, maybe burst release or crystallization type of the problem during the storage conditions. If we prepare some of the emulsion, but the emulsion mostly have the poor stability, and microencapsulations have the heat sensitive type of compound may be degrade by this microencapsulation type of the methods. So by this literature review, we can conclude that we should find some new method or technology to give or to improve the bioavailability of the vitamin D in our body. Here the figure for challenges in oral bio, why the oral bioavailability of vitamin D is lower. There may be some of the chances like first pass metabolism or co-required solubility, maybe inappropriate partition coefficient, the insufficient time of absorption. So here, by this literature review, in comparison to the existing innovation technology, the supersaturated SNAD, that is super saturated self-nano emulsifying drug delivery system, offer this type of the advantages. First, if we prepare the SNEAD type of the formulations for vitamin D, SNEAD mainly do not contain the water. So therefore, chemical and physical stability of the system is higher, and we can directly give our dosage form in a single unit of type in hard or soft gelatin capsules or maybe in HPMC capsules. Then we can administer the dose in this type of system from 25 milligram to greater than 2 milligram. So patient compliance is high. Then the enhance the important property of vitamin D supplementation like therapeutic efficacy, photostability, biodegradation. Then they improve absorption and bioavailability by reduce the necessary of lipids in absorption, then provide the protection against the harsh environment of GIT, and also enhance the transmucosal type of the transports. So now here the super saturated
Madam, you
SNEADs.
can go to your results. You please share your results.
Okay, ma'am, but I present my review work on this topic.
Okay.
Okay. And
Okay,
super saturated SNEADs, okay, ma'am, are feasible to manufacture also and easily can be scaled up also. Here you can see how it will be work. When we take the super saturated SNEAD by the capsule form in the HPMC capsules by the gastric motility, it will convert into nano emulsion. Then by this nano type of the emulsion, that will contain mainly oil surfactant and co-surfactant. And here we can see the HLB range of these different type of the excipients that might be used for this formulations. Here, mainly the ripple formulations are mainly four types: type 1, 2, type 3A, type 3B, and type 4. In type A, mainly lipid contents are 100%, type 2 contain 40 to 80%, the surfactant are 20 to 60%, and mainly the SNEAD type of the system that will be in type 3B that contain lipid less than 20%, the surfactant more than 20 to 50%, and hydrophilic co-solvent are 20 to 50%, and the particle size may be less than 100 nanometer. Here are some examples of lipids like cape mule, peanut oil, coconut oil, olive oil, surfactants like 20, 40, 60, 80, poloxama, cremo for EL, then co-solvents PG 600, 400, ethanol, transferal HP that can be used for this type of the formulations. In that, we can, after this selection of these excipients by their solubility measurement, then we can construct the ternary or pseudo-ternary plot type of the diagram. Then we can select the excipients further by percentage transmittance type of this study, and we can go further by optimization of these formulations, and we can go further with in vitro evolution and then in vivo evolution. Here are the tools and technologies to be used to solve these problems. In that first way, the polymer, the precipitate inhibitors type of the polymer, because we are going to prepare a supersaturated SNIDs. So in that, first take the polymer precipitate inhibitors, then take the oil surfactant, co-surfactant, and homogenizing by heating by the mild temperature by the vortex mixing. Then you can get the blank SNIDs. In that, you can add your drug, excess amount of the drug. Then SNID and excess vitamin D mixtures are shaken at 100 RPM for 24 hours by staking water bath. Then mixtures are centrifuged for 15 minutes and the supernatant are separated, and you will get the final supersaturated SNIDs. Here the mechanism of self-nanoemulsification by the diffusion, and how the SNID system that will be absorbed through by intestine. In that, when you take the SNID system by the gastric motility or by the gastric agitation, it will convert into the nanoemulsion. Then that will secrete or the bile salt, and by this bile salt, the further emulsification of these oil droplets with the drug, and then the triglyceride of this oil content will convert into the di or monoglyceride in the presence of cholesterol or phospholipids and lipase, and then this monoglyceride will be absorbed through the intestine into the lymphatic system. Here the demo presentation, how we can prepare this SNID into the lab scale. Now the advancement in self-emulsification system, that the main title of this abstract of review work, that is supersaturated SNIDs. The in general, in simple SNID, we just used a high concentration of hydrophilic type of the surfactant, but that might cause the toxic effect in our body. And also the chances of drug precipitation or crystallization during the in vitro study. So we can use the polymeric precipitation type of inhibitors, that certain type of polymers like HPMC, PVP, or Solute Plus, polyvinyl acetate, polyethylene glycol, that work as a PPI, that is polyprecipitation inhibitors, and that produce the supersaturated state in vivo, and that encircle the droplets with hydrophilic nature, which overraise the oswald ripening and act as a PPI, and that is various grades of this type of the polymers like HPMC and the PVP. Here the mechanism of precipitation inhibitors. If we prepare just simple type of SNID that work as a spring type, and if we use a PPI.
Sorry for the interruption, ma'am. Try to wrap up with the presentation. The time is up.
Okay. Thank you.
Is there any tablet that is available in the market prepared using this technology?
Yes, ma'am. Can you please?
Is there any tablet available in the market prepared using this technology?
No, ma'am. Not available right now.
Are you sure?
Ma'am, actually, the SNID type of this system is available, but not available. The supersaturated SNIDs are not available.
Okay.
Thank you.
Sure.
Okay, ma'am. You can stop sharing your screen. We'll move on with the next presenter, Ms. Simran Kaur from Christ Deemed to be University, Karnataka, India for her oral presentation. Ms. Simran, I hope she's not in the Zoom. I'd like to call our last presenter, Lata Kotapalli, who has missed her presentation. Is she here? Lata Kotapalli? Okay. The next presenter would be Mrs. Manisha Nitin Veer from Bharati Vidyapreet College of Pharmacy for her oral presentation.
Yes, ma'am. Ma'am, am I audible?
Yes, ma'am, you are audible.
Okay, ma'am. Ma'am, my screen is visible?
Yes, ma'am, kindly put it in the full screen mode.
Yes, it is in full screen mode, ma'am.
Actually, here we can't see. Okay, ma'am, you can continue, not a problem.
Okay. Okay, so good morning, everyone. Myself, Mrs. Manisha Veer. I am working under the guidance of Dr. Mrs. Neela Bhatia, ma'am. And today I am presenting synthesis and characterization of raloxifene conjugate for breast cancer therapy. These are the contents of my presentation. So as we all know, breast cancer, it is the leading cancer in women across the world, and it is considered as the second chief mortality cause in women. There are various treatments available for the breast cancer. Depending upon the stage at which the cancer is diagnosed, various treatment approaches employed for the breast cancer management. The management of breast cancer requires proper monitoring and optimized treatment schedules in the patient. The various chemotherapeutic agents which are used in the treatment of breast cancer, one of them is raloxifene. The rationale of this study is raloxifene, it belongs to selective estrogen receptor modulators and it is used for treating osteoporosis. Recently, FDA has approved raloxifene for reducing the risk of invasive breast cancer in postmenopausal women with osteoporosis and in postmenopausal women at high risk of invasive breast cancer. Raloxifene has two hydroxyl moieties and rapidly conjugated by phase 2 metabolism pathway, which makes it difficult to achieve the adequate bioavailability on oral administration. So here in the present work, we have synthesized ester conjugate of raloxifene with non-steroidal anti-inflammatory drugs so as to improve the cytotoxic effect of raloxifene along with additional analgesic anti-inflammatory activity. This conjugation will temporarily mask the hydroxyl groups which are present in the raloxifene because as we know, the hydroxylated compounds are rapidly undergo the phase 2 metabolism. So this will help to improve the cytotoxicity of the raloxifene as well as the another moiety which is non-steroidal anti-inflammatory drug which will provide the additional analgesic anti-inflammatory effect. So let us first see the drug profile. So raloxifene, it is a yellow solid. Molecular weight of raloxifene is C28H27NO4S. The mechanism of action of raloxifene is as it is a selective estrogen receptor modulator. In some tissues, it acts as agonist, whereas in some tissues, it acts as antagonist. The tissues like bone,
Slides
liver,
are not
in which...
moving.
Yes, ma'am?
Your slides are not moving. It's still in the first slide.
Now?
No.
Rationale of the study, ma'am, is it visible to you?
No, no, no.
No?
You can go to your result and discussion.
Ma'am, is it moving now?
No, it's still on the first slide. You can see only the first slide.
Can anybody help me? What should I do?
You select the slideshow mode.
Yes, ma'am. On my screen, slides are moving, ma'am.
No, slideshow. You select the slideshow mode.
Hello, ma'am.
Yes, ma'am.
Yes, in the corner, can you see one slide share screen is there? So you go to there, and in the corner there is a slide
No,
screen.
ma'am. Corner is not showing
Uh,
any option here. In more option, ma'am.
No. In the in the top corner there is slide screen share is there. Your screens are not visible now. In the corner there is slide screen share. You go to and click just that. It is coming on mode. Otherwise, you can give share your screen to other people. They can operate this also.
Uh,
Sorry, ma'am, is there an issue?
just a moment. May I just help the presenter as to how to go about it?
Yes, ma'am, you can.
Okay, perfectly all right. Do you know how to minimize the screen? Can you minimize your screen, the Zoom screen?
Are you using laptop or mobile?
Ma'am, it is laptop.
Okay.
Then press F5. F5.
Okay.
Functional key F5.
Yes, ma'am.
You minimize your screen now?
Ma'am, now it is visible, na?
The slide was visible, but it is not changing. Is this the case? Your slide is visible to us.
Okay.
Okay.
But it is not moving.
Are you changing it? Are you
Yes.
tabbing it?
Yes, I have changed it to the next slide, but it is not visible to you.
On the screen. Right. I'll tell you what to do. Now, what you do is you just on top of the Zoom screen, you'll see there will be three bars. One will be for minimizing it. The
Ma'am,
other
your voice is not clear. Hello.
You just minimize your Zoom screen. It will be minimized to a little block, a rectangular block. Are you able to do that?
Ma'am, I hope she has been shared only her PPT screen, not her full screen. 1 minute, let me guide her. Manisha, can you just stop sharing your screen?
Okay, I will stop.
Yes.
Yes, ma'am.
Okay, now give share screen. Once you give this share screen, you will be seeing share entire screen. Yes, now we can see your Reloxifin. Yes. Can you, if you are not able to put in full screen, that will not be a problem. Just click the down arrow and make sure that the 5th slide is changing to 6th slide, 7th slide likewise. In the slide column, can you see the downward arrow at the bottom? Click add notes, you can see, right?
No, ma'am.
Ma'am, in the left hand side, right bottom, left hand side. 5th slide, 6th slide, you can see?
No, ma'am.
Ma'am, you no need to make full screen. Just go to the PPT screen itself. Close your full screen. Give escape and close your full screen.
Yes, ma'am.
Now you are in the content slide, right?
Second slide, yes.
Yes, go to the introduction slide. Yes, like that itself you make the presentation. That will not be issue.
Okay, ma'am. Okay, thank you, ma'am. Okay. Okay, so introduction.
Move to result and discussion.
Yes, ma'am. Shall I start, ma'am? Hello.
That's what, ma'am. You can switch over to result and discussion. No need for introduction.
Okay, ma'am. Okay, so here is the method of synthesis of this raloxifene NSAID conjugate. We have synthesized series of raloxifene NSAID conjugates. Out of that, here I'm going to present only one conjugate, which is of raloxifene and aspirin. So here first, NSAID, that is aspirin, is converted into the acid chloride, and then it is coupled with the raloxifene. So as to form this raloxifene aspirin conjugate, which is a ester conjugate, and it is denoted as M1. This is the structure of raloxifene aspirin conjugate. Physicochemical properties of the synthesized conjugate given here on the slide. So the product confirmation of the synthesis of product was done by the TLC, and where we have used the cyclohexane ethyl acetate mobile phase in the ratio of 1:1, and these are the RF values of all the three components, that is raloxifene, aspirin, and MV1, which is the synthesized ester conjugate. Structural confirmation was done by the IR, NMR, and mass. This is the IR spectrum of MV1 conjugate. Here we can see the absorption band at 1682 for CO stretch. This indicates the formation of ester, whereas this 1173 absorption band is showing the CO stretch of the carboxylic acid ester. Proton NMR also was taken for the synthesized conjugate, and it has confirmed the number of protons which are present in the conjugate, that is 76 protons are there. C13 NMR was taken for confirmation of the number of carbons in the synthesized MV1 conjugate, and it has confirmed the total carbons which are present in the conjugate are 46. Mass spectrum shows that the molecular ion peak at 797, that is the molecular weight of synthesized components. So in this way, the structural confirmation was done by taking the IR, NMR, and mass spectrum. Solubility studies were done for the synthesized conjugate MV1 in different solvents. The conjugate was highly soluble in methanol, followed by the ethanol, chloroform, ether, acetone, and having very less solubility in the water. Partition coefficient was determined for the synthesized conjugate, and it shows the non-polar nature of the conjugate. HPLC method was developed for the synthesized conjugate, and the method was validated as per the ICH guideline. These are the details of chromatographic method developed for the MV1 conjugate. We have used this methanol, water, 0.1% trifluoroacetic acid in the ratio of 60:15:25 in the ratio of volume by volume, whereas the retention time observed for all three components was for raloxifene 9.65 minutes, aspirin 11.07, whereas for MV1 20.97. The method was validated as per the ICH guidelines, and this table shows the superior accuracy of the developed method, whereas the method has shown the good linearity with R-square values ranging from 0.999 to 1. For the determination of hydrolysis of the conjugate, after the validation of HPLC method, the kinetics of hydrolysis of synthesized conjugate ester was studied at various pH, that is 1.2, 5.5, 6, and 7.4, as well as in the simulated gastric fluid and simulated body fluid. The results showed that minimum amount of the conjugate hydrolyzed at gastric acid pH, as well as in simulated gastric fluid and simulated body fluid, and at the physiological pH 7.4. However, the conjugate rapidly hydrolyzed in the plasma, which suggests that the activity of esterase enzyme present in the plasma, which acting on the conjugate. The reaction kinetics for degradation of the ester conjugate to its parent compound was first order. Further, the cytotoxicity of the synthesized conjugate was assessed by in vitro cytotoxicity assay using the MCF7 breast cancer cell lines, and it was found that the conjugate more cytotoxic than free raloxifene in MCF7 cell lines, indicating that the improved potential of the conjugate to treat the human breast cancer. The anti-inflammatory activity was assessed by in vitro anti-inflammatory activity using this protein denaturation method, and the synthesized conjugate showed better anti-inflammatory activity than the standard aspirin drug. So it can be concluded that the synthesized conjugate MV1, that is raloxifene aspirin conjugate of the parent drug raloxifene, may prove to be applicable prodrug in reducing the systemic toxicity of raloxifene, as well as selectively targeting the cancerous cells and additional analgesic and anti-inflammatory effect. By this means showing fulfillment of the conjugate design. These are the references. Thank you.
What was the pH of the mobile phase you have used in HPLC?
Yes, ma'am.
pH of the mobile phase, what you used in HPLC?
Mobile phase, it is methanol water.
No, what is the pH? pH I am asking. What was the pH?
Okay.
But you have 4 peaks, right?
Ma'am, we have also introduced the salicylic acid because the NSAID which we have used is aspirin. So there might be chances of release of salicylic acid from that aspirin. So this last peak is of salicylic acid. So in case if in vivo study is carried out, if salicylic acid is going to be released, then that can be identified. So therefore we have introduced salicylic acid also.
Okay. Yes, we can move on to the next presentation.
Yes, ma'am. Okay, we'll
Okay,
move on with
thank
the
you,
next presentation. It is the call for the missed out presentation. Simran Kaur, are you there? Simran Kaur? Dr. Lata Kotupalli? Okay, the next presenter will be Shakti Priyadarshini from SRM College of Pharmacy, SRM Institute of Science and Technology, India, for her oral presentation.
Good afternoon, ma'am. Am I audible?
Yes, ma'am, you are audible.
Madam, my screen is...
Yes, ma'am, your screen is visible, yes.
Yes. Thank you, ma'am. Is my slide changing, ma'am?
Oh, yes, ma'am. Yes, it is changing. You can present. You can start.
Good afternoon, all. I'm Dr. S. Shakti Priyadarshini. I'm working as an assistant professor in the Department of Pharmacology, SRM College of Pharmacy. My topic is on GCMS analysis and in vitro alpha amylase inhibitory activity of leaf extracts of Ravenella madagascariensis. Diabetes mellitus is a chronic condition that is characterized by raised blood glucose levels. It is the most common endocrine disorder, usually occurs when there is a deficiency or absence or impairment of insulin activity. People with diabetes are also in the increased risk of cardiovascular, peripheral vascular, and cerebrovascular diseases. The present study was undertaken with a view to document the GCMS profile and in vitro alpha amylase inhibitory activity of Ravenella madagascariensis sun leaves. The literature reviews are presented here. The pharmacognostical standardization was carried out by Shakti Priyadarshini et al. And the epidermal structure of leaf was recorded by Trivedi V.S. et al. The phytochemical studies include bioassay-directed fractionation and phytochemical evaluation of seed areas of Ravenella madagascariensis. The pharmacological studies reported in the plant includes the in vitro insulin secretagogue effect and the antifungal and toxicological activities. Materials and methods. It includes, we started with the extraction of the plant material, followed by histochemical analysis, GCMS analysis, and in vitro alpha amylase inhibitory activity. Extraction was carried out by successive Soxhlet extraction. Here, the coarsely powdered leaves of Ravenella madagascariensis were treated with solvents of increasing polarity by Soxhlet percolation. The N-hexane and ethyl acetate are the solvents used. The extract was dried and it was weighed and the percentage yield was calculated. The N-hexane extract showed a percentage yield of 0.75% weight by weight, whereas the ethyl acetate extract showed a percentage yield of 4.38% weight by weight. Further, the transfer sections were treated with the different chemical reagents and the presence and the localization of phytoconstituents were recorded. Tannin was found present in the epidermis, the mesophyll, cortical cells, and parenchymatous tissue. Saponins were found in the midrib parenchyma. Fats in the mesophyll and phloem parenchyma, and alkaloids were distributed in the mesophyll, midrib, upper and lower epidermis, and parenchymal cells in midrib. GCMS analysis was carried out by Agilent 7890B, 5977A, sorry. Not see the screen here. How to close this one? So I could see on my screen. Excuse me, ma'am. Okay, so the sample 1 microliter was used as a sample. Helium was used as a carrier gas at temperature 280 degrees centigrade. The GCMS profile of my extract showed around 20 compounds in the N-hexane extract, including the gamma tocopherol, phytol, gamma tocopherol ester, triaconthane. The ethyl acetate extract showed the presence of tetratriaconthane, gamma tocopherol esters. Alpha amylase inhibitory effect showed that among the two extracts, the ethyl acetate extract showed the higher alpha amylase inhibitory activity with an IC50 value of 91.75 microgram per ml in comparison to the standard acarbose, which showed an IC50 value of 78.72 microgram per ml. Thus, we conclude that the results of the present study indicate that the volatile bioactive components were present in the N-hexane and ethyl acetate extract, which were reported with various biological activities. And the among the two extracts, the ethyl acetate extract was shown to be having the higher alpha amylase inhibitory activity, aiding in the lowering of postprandial glucose levels. And further studies can be put forth in the design and development of Ravenella madagascariensis leaves and its extracts into a suitable antidiabetic formulation. These are the references. Thank you.
What is the common name of this plant?
Ma'am, Visiri Varai, ma'am. In Tamil, we call this. And in English, we call it as a traveler's palm.
Okay, either you have used leaves or old plant.
No, ma'am, leaves were used.
But your heading and conclusion shows it's leaf extract. Whereas yes, your results you have mentioned a extract from whole plant extract. You check in the results you have mentioned as whole plant. That's why I have that.
Results, ma'am.
Yes. Where did you perform your GCMS?
In the SRM College of Pharmacy. We have a department of biotechnology.
Okay. When your results you have mentioned as whole plant, you'll be clear with what you have done, whether leaves or whole plant.
Okay, ma'am. It's leaves, ma'am. We did the extraction with the leaves.
That's what you need the correction in your results. Okay, thank you.
Thank you, ma'am.
Thank you, ma'am. We'll move on with the next presentation. Ranchana R. Eligar from Sri Ravatpukar Sarkar College of Pharmacy, Raipur, India.
Yes, ma'am.
Yes, ma'am, you can start with your presentation.
Just a second.
Ma'am, can you share your screen?
Sure. Yeah, just a second. Hello?
Yes, ma'am.
That virtual background is coming.
No need of virtual background, ma'am. Just share your screen and go to the PPT. No need of virtual background. That will not be issue. Make sure you have opened your PPT in the other screen. Then come to the Zoom. Give share screen. Ma'am, can you share?
Yeah, just a second.
Can I guide you step by step? Yes, okay, fine.
It's visible?
1 minute. Yes, you can start now.
Yeah, myself Rachna Ragar, PhD scholar from Rawatpura Sarkar College of Pharmacy, Raipur, Chhattisgarh, India. I'm going to present a topic on box bank and design approach to develop and optimize genistein conjugated transferasome and mannose conjugated genistein transferasomal gel. Here are the few facts about skin cancer. Genistein is mainly my work is mainly on the skin cancer. So few facts about skin cancer. Once in every 1 hour there is a death due to skin cancer. This is how the skin cancer starts, and initiation and malignancy tends to. Melanoma is developed due to many factors, that is genetics, age, UV radiation, nuance, and degree of skin pigmentation. This is how it develops. It starts from the squamous types of skin cancer are squamous cell carcinoma, basal cell carcinoma, and melanoma. Now coming to genistein. Genistein is a nutraceutical soy isoflavone, plant estrogen found in soybeans. Its drug profile, molecular weight is around 270.24 gram per mole. Physically, it is a solid. Partition about 0.84. Biological half-life is about only 3.2 hours. Solubility, it's only soluble in organic solvents due to higher molecular weight. Based on the pharmacological classification, genistein falls in phytoestrogens, anti-carcinogenic, and protein kinase inhibitors. Mechanism of action, mainly protein tyrosine kinase disrupt the signal transduction and induce cell differentiation. Inhibition of topoisomerase 2. These are the two phases, two types where the genistein mainly acts. These are the benefits of genistein. Mainly, delivery of genistein is affected due to poor water solubility and hydrophobic, reduced permeability, and therefore low bioavailability. In order to combat this, so there are many options, that is liposomes. With the help of nanotechnology, we can develop liposomes, polymeric nanoparticles, micelles, metals, and nanocapsules. Here, my topic is on objective is to develop the genistein treated transferasomes. Work plan starts from P-formulation studies, formulation development, characterization, and the biological characterization followed by. These are the advantages of nanotechnology. This is mainly the transferasome, structure of transferasome, where the drug is in the transferasome incorporated. And this is the ligand conjugated transferasome. This is how transferasomes acts through the transferasomes, mainly penetrates deformation and followed by reformation. Pre-formulation studies are procurement, then partition coefficient, selection of different pH media, and compatibility studies. Formulation development. Box bank, and next is the box bank and design. Box bank and design is a 3-square factorial design where formulation variables, variables are phospholipid and surfactant ratio, sonication time, and RPM. Then responses are vesicle size, percentage entrapment efficiency, and percentage drug loading. Based on this, calibration curve was prepared. obtained. The concentrations were 5, 10, 15, 25 mg. This is the melting point, genistein, where 302 was the melting point of genistein, mannose, and followed by the soy lecithin, that is the phospholipid used. This is the FTIR spectra of the genistein, physical mixture, genistein, mannose, and soy lecithin. UV spectra analysis, it showed the maximum lambda max at 259.8 nanometer. Depending on the particle size determination, it was 182 nanometer particle size. Polydispersibility index was 0.268 and results were too good and acceptable. Zeta potential was found to be minus 27.1. This is the TEM structure where the particle size range was 200 nanometer range. That's all to present. Conclusion. The study revealed that both genistein transferosomal gel and mannose conjugated transferosomal was successfully optimized by the Box-Benton design. FTA study shows no incompatibility. Both the gel physical characteristics were acceptable and good. Mannose conjugated genistein transferosomal formulation showed better results. Thank you.
Is it your preliminary data or because many other characteristics are not shown?
Just preliminary studies, ma'am, just physical characterization done, waiting for the main data.
Okay. Okay. So what for you have done FTIR studies?
Yes, sir. For melting point.
FTIR.
Yes, ma'am.
Why did you perform FTIR?
To study the compatibility.
Okay. Okay. You can stop sharing so that the next person can listen.
Okay, ma'am. Thank you.
Thank you, Ranjana.
Thank
We
you.
move on with the next presenter, Ms. Swati Vijaya from PES University for her e-oral presentation.
Good afternoon, ma'am. Can I present my screen?
Yes, ma'am, you can.
Now is the screen, is the present PPT available?
No, not it. It is not yet visible. Your screen you are sharing, but we can't see your PPT. Yes, now we can see. Yes, proceed with your presentation, please.
You are not audible. Swati, you are not audible.
Ma'am, can you able to hear me now?
Yes.
Ma'am?
Please share your screen. You are audible now. Yes, yes, please
Is
proceed.
it visible? Ma'am, is it visible? So,
Yes, ma'am, it is
Yes, yes.
visible. Abstract is visible.
the objective of the proposed study are as follows: that is, the pre-formulation studies, including the identification, characterization of the lamotrigine was carried out, and the preparation of the powder blend of the drug and excipient was done, and the pre-compression assessment of the powder blend, such as angle of repose, bulk density, Cars indexity, and Horsner's ratio was carried out. And next is the need for the study. So, need for the study, the demand of orally disintegrating tablet of the lamotrigine has been growing during the last decade, especially for the pediatric patients and also for the dysphagia condition. Lamotrigine has a longer half-life period that ranges from the 22 to 37 hours. Therefore, it shows a very low therapeutic action. It has a main drawback in the bipolar and the maniac attack. By formulating the orally disintegrating tablet, this drawback would be overcome. The marketed extended release tablets show a very slow disintegration rate and dissolution. In the medicine, it takes in the gastrointestinal tract. In doing so, they extend the time that it takes for the active ingredient to be absorbed into the circulatory system. Next is introduction. So epilepsy is a disorder of the brain characterized by the repeated seizures. A seizure is usually defined as a sudden alteration of the behavior due to the temporary changes in the electrical functioning. What are the causes is that head injury, brain tumors, cerebrovascular disease, infectious etiology, alcohol, stimulant drugs, and others. So we are formulating the orally disintegrating tablet of lamotrigine. So it is a solid dosage form containing the medicinal product which disintegrates rapidly, usually within a few seconds, which placed upon the tongue. So the mechanism of action are as follows: the water must quickly enter into the tablet matrix to cause a rapid disintegration. The incorporation of the appropriate disintegrating agents or highly water-soluble excipients should be there in the formulation. There are some other mechanisms that have to be followed, that is high swellability of a disintegration, chemical reaction, and the capillary action. So the basic approaches to develop this ODT is, it includes the maximizing the porous structure of the tablet matrix, incorporating the appropriate disintegrating agent, and using the highly water-soluble excipients in the formulation. So next is the materials and method. So the drug uses lamotrigine, the lubricant is magnesium stearate, and the super disintegrant in the Pharma Best 500, cross-povidone, and the cross-caramelose sodium. Anti-caking agent is silica dioxide, and the binder is avicin pH 1 or 2, that is microcrystalline cellulose. Sweetener used is mannitol. So the drug profile, it comes under the BCS class 2. It is used for the anti-epilepsy. The class is phenyltrizine. Its appearance is white color amorphous powder. The partition coefficient is 5.2. Protein binding is 50% to 60%, and half-life is 22 to 37%. So the pre-formulation studies which was carried out, it says that it is soluble in the ethanol and insoluble in the water. The melting point was at the range of 217 degrees Celsius. Next is excipient profile. So the very first super disintegrant is Pharma bus. The composition is 95% of fructose and the 5% starch. There's a rapid disintegration time, and it produces the mouthful qualities. It has a highly compactable nature that carries the drug loads without increasing the tablet size. Next super disintegrant is cross-povidone. It is white, free-flowing, odorless, and tasteless with hygroscopic property. And synonym is polyvinyl pyrrolidone. So cross-povidone particles are porous and granular in nature. It will enhance the wicking of the liquid into the particle. Next is cross-caramelose sodium. The physical property is it is insoluble in the anhydrous ethanol. The synonym is carboxymethyl ether. It acts as a super disintegrate agent due to its high affinity to the water absorption. It is effective at the lower concentration. Next is method of preparation. Lamotrigine, the orally disintegrating tablet, were prepared by direct compression method. All the ingredients were sieved through 60 mesh sieve separately and collected. The drug and other ingredients were waved and mixed in the geometrical order. The resultant mixture was directly compressed into the tablet by using a 7 mm round flat faced punch on the rotary tableting machine. So 200 mg tablet was formulated. So we have formulated 7 formulations. In that, the lamotrigine drug is 25 mg, which is fixed for all the formulation, and the magnesium stearate is of 2.5. And mannitol, the sweetener agent, and microcrystalline cellulose, silica dioxide. These are the excipients which have been used. And the formulation 1 to formulation 3, the Pharma bus 500 is used. 4 and 5 cross-povidone was used, and 6 and 7 cross-caramelose sodium is used. The evaluation parameters was carried out. There is pre-compression studies. It involves the bulk and tab density, Cars index, Housman's ratio, angle of repose, and FTIR studies. The post-compression studies involve the friability, hardness, weight variation, drug content, and in vitro drug release, in vitro disintegration time. So next is first is pre-compression studies. So the bulk density is a ratio. Bulk density is the ratio of the weight of the powder to the volume of the powder packing. So it ranges between the 0.36 to 0.45. And the tab density, it is the ratio of the weight of the powder to the tabbed volume of the powder. So this was carried out mechanically tapping the graduated cylinder. So it ranges between the 0.42 to 0.59. And the Cars index, Cars index was calculated by using the bulk density and the tab density. And the F3 formulation shows the very good Cars index value. Housman's ratio, it is another parameter to characterize the powder. And it ranges between this 1.10 to 1.51. So it shows a very good, excellent flow property for all the formulation. Next is angle of repose. The formulation 3 shows 20.80. That is a very excellent, it shows the excellent flow property. And F1 and F2 formulation shows a good flow property, and remaining F4 to F7 shows the moderate flow property. Next is FTIR studies was carried out. From the above graph, the peaks representing the pure drug were similar in all the graph, suggesting that there is no such interactions and the pure drug is not altered functionally. The FTIR. The FTIR. Sorry, ma'am. The FTIR spectra of the drug and the super disintegrant are not having any significant interactions. And next is post-compression study that involves thickness, hardness, and weight variation, friability, and the drug content. The thickness and the hardness was.
Other than the presenters, others, please mute. Ruby Manjari, please mute your audio.
The thickness.
Swati, you can go to the last slide.
Okay.
What are these plus and minus values?
Ma'am?
what are these plus and minus values? Everywhere you have represented plus and minus. What are those values?
Ma'am, this is a parameters which we have conducted for thickness, hardness, and weight variation.
That I understand. You have given a value plus or minus another value. What do you mean by that?
Ma'am, in between the range, the value is present, ma'am.
That's it. What is it called?
Sorry?
Okay, you can move to your last slide. Okay.
Conclusion, it is, ma'am?
Yes, you proceed with this.
It is concluded that the fast dissolving tablets of lamotrigine can be successfully prepared by direct compression technique by the fast disintegrating excipient in order to improve the disintegration and dissolution of the drug. The present study indicated the positive results to prepare the orodispersible tablets of lamotrigine by direct compression method using the 3 different super disintegrant. F3 and F2 formulation has shown the faster disintegration with F3 also shows the exhibiting better in vitro drug release. Hence using F3 data, pilot plant scale-up can be carried out. The future perspective is studies on the solubility enhancing technique followed by animal experiment on formulation shall provide a valuable safety profile. Comprehensive trials with the pilot plant scale-up volume will progress the data. Thank you, ma'am. Ma'am, can you able to hear me?
Yes, madam, you can stop sharing. We can move on to the
Yes, ma'am, we can move on with the next presenter. I'd like to call Simran Kaur from Christ deemed to be University, Bangalore, Karnataka, India. Simran, are you there?
Yes, ma'am.
Yes, you can start with your presentation.
Sure, ma'am. Ma'am, it says to quit and reopen for sharing. So I will be joining once more.
Sorry?
Ma'am, my system is saying to share the screen, I need to quit and reopen the meeting.
Okay, fine. Just try joining again. We'll move on with the presenter Chloe and we will do the presentation next. Okay?
Okay, sure, ma'am.
Okay.
Good afternoon, ma'am. Can you hear me?
We'll move on with the next presentation. Kohli Pranjali Bajrang from Christ University for her EOB presentation.
Ma'am, is my screen visible?
1 minute. Yes, it is visible. Can you make it full screen? Is it possible?
Yes, ma'am. I'm making it. Myself, Sole Pranjali Bajrang. I am a research scholar from the Christ University, Bangalore, Karnataka. Today, I will be presenting my work, Green Synthesis of Cobalt Oxide Nanoparticles from the Punica granatum L and its pharmacological applications. So, first of all, Punica granatum, pomegranate, is a fruit-bearing deciduous.
Screen is not changing actually. You
Ma'am,
have not...
and second slide.
No, it is there still in the first slide. Just give escape and proceed with the PPT screen itself. No need of full screen.
Okay, ma'am. Now, ma'am.
Yes, you can do it. Yes.
Okay. Punica granatum is a fruit-bearing deciduous plant belonging to the Litheraceae family, and pomegranate is well-known medicinal plant in traditional medicinal systems like Ayurveda. It is one of the proven source for antioxidant, anti-cancer, anti-diabetic, anti-inflammatory, and antimicrobial activities, depending upon the ethnobotanical uses that are proved by the researcher for pharmaceutical application. Liquid extract of seed shows the antibacterial and antifungal properties against the Salmonella and Penicillium species, and pomegranate seed oil has shown advantageous effect as an anti-cancer agent and in the treatment of colon cancer and breast cancer. When we did the survey about the nanoparticles, we found that by using the various parts of pomegranate like leaves, peel, seed, juice, and fruit extract, silver nanoparticles, iron nanoparticles, gold nanoparticles were synthesized, which showed antimicrobial, antioxidant, and anti-cancer activity against different pathogens. As there is no report available on the green synthesis of pomegranate seed oil, this study will help to remove this gap and promote the green synthesis with different oil samples. So on similar basis, the main aim of this study is to highlight the application of Punica granatum seed oil to synthesize the cobalt oxide nanoparticles by using the green synthesis method and to evaluate its efficiency in pharmacological properties like antimicrobial and in vitro cytotoxicity studies. So for that, we use materials and methods, which is like, first of all, pure pomegranate seed oil we purchase from ICAR NRCP, Solapur, Maharashtra. And for isolation of pomegranate seed oil, they use the cold pressed extraction method. With that, we use 0.1 molarity of cobaltous chloride solution and distilled water. So here is the diagrammatic view of synthesis of cobalt oxide nanoparticle. So pomegranate seed oil was mainly used as a reducing agent, whereas cobaltous chloride solution was used as a precursor agent in the green synthesis. And this reaction mixture we kept for the 1 hour constant stirring at temperature 45 degrees centigrade. And once this reaction is finished, after that we wash the particles and we dried it, and after that we proceed with the characterization of cobalt nanoparticles, which are like UV spectroscopy, XRD, DLS, scanning electron microscopy, and FTIR. So when we gave the results, first in first A image, we can see initial color of the reaction mixture was completely dark green, and in B it was light, it became a light green. And after that, in C image, image C, we have kept the UV visible spectrum for cobalt oxide nanoparticle shows peaks at for cobalt chloride it showed 279 nanometer, and for precursor salt, which one we use for that, it showed the 510. So this study proves that the cobalt chloride particles had reduced during the synthesis of cobalt nanoparticles. The resultant product showed different surface plasmon resonance area than the precursor salt. And this proves that the green synthesis of cobalt has been successfully done with the reduction of cobalt salt. That's why we proceed further for the optimization of reaction condition. For that, we use the DLS, and DLS mainly we use for the size analyzing, and optimization of reaction mixture was done with different conditions like concentration of oil and temperature. The different trial and error methods we perform with the different concentration of oil, that is 3%, 5%, and 7%, and different temperature 30 degrees centigrade, 35 degrees centigrade, and 45 degrees centigrade. But at 5% of concentration of oil and 45 degrees centigrade of temperature, the average particle size of cobalt particle was 129 nanometer, and all results are shown here in the given figure number 3. Once we got the size 129 nanometer, we further did the XRD, and XRD it was like graphical pattern what we received for the synthesized cobalt oxide nanoparticles. So observed pattern, we observed that pattern, and we analyzed we compared with the different cobalt particle pattern, and it was matched with the green synthesized cobalt oxide nanoparticles. Hence, depending on the observed pattern, it is proved that the green synthesized cobalt nanoparticles are cobalt oxide nanoparticles. Further, for scanning electron microscopy and the size and morphological texture and characters for nanoparticle, we use the scanning electron microscopy, and it is observed that cobalt oxide nanoparticles were in spheroidal shape with smooth texture and uneven in number, and each of nanoparticle size was ranging below the size of 100 nanometer. Further, we did the FTIR, Fourier transfer infrared spectroscopy, and in that functional groups like metal carbonyl and alkyl was found. After that, we proceed with the antimicrobial activity. Firstly, we did the antibacterial and antifungal studies. So in that, in vitro antibacterial and antifungal activity of green synthesized cobalt oxide nanoparticles were studied by using the agar-well diffusion method against different disease-causing microorganisms. So for antibacterial, we use 5 different bacterial strains, and for antifungal, we use candidiasis different strains, fungal strains, and in that chloramphenicol and fluconazole we use as a standard drug for in antibacterial and antifungal activity respectively. From the results and from the table, you can see that here Pseudomonas aeruginosa and Bacillus subtilis shows the highest zone of inhibition, which is near about 27 millimeter. Okay, ma'am. Here is a in vitro toxicity of the cobalt oxide nanoparticles, where MCF7 brace cell line we have used as a cancerous cell, and L line 29 cell we have used as a non-cancerous cell, and the IC50 value for MCF7 was 57.85, and here is a comparative analysis for in vitro toxicity, and here becomes and for non-cancerous more than 1000 microgram per ml at concentration also there was only 70% of the cells were live. And we comes to the conclusion. This study concludes that synthesis of cobalt oxide nanoparticles from Punica granatum seed oil and its potential in pharmaceutical industries as an antibiotic agent. Meanwhile, this study helps to remove the gap in green synthesis with oil samples. Various human pathogens like different bacterial and fungal pathogens, which are responsible for various infections like candidiasis, can be controlled by using these nanoparticles, and these nanoparticles can be used to treat the cancer. It strongly proves that the cobalt oxide nanoparticles will not show any harmful effect on normal human cells. Hence, nanoparticles can be used in the form of ointments, plasters, first aid creams, bandages, and emulsions form for the external use. Going forward, this study can be very helpful to medical research centers and pharmaceutical industries, as well as to deal with the modern problems of mankind, like increasing antibiotic resistance in the community. These are the references which I have used.
Okay, ma'am. So what is the major problem when you use cobalt?
Cobalt, ma'am, usually people have myth like cobalt is a poisonous for human being, but we have proved that cobalt we can use as an antimatter.
Perform the long-term toxicity, right? Acute toxicity and chronic toxicity. Okay, ma'am, we can move on to the next.
Real and anti-cancer agent.
Okay.
Thank you so much. Simran Kaur, are you ready?
Yes, ma'am.
Yes. Yes, you can start with your presentation.
Yes, ma'am.
Okay.
Ma'am, is my presentation visible?
Yes, it is visible.
Okay. So, good afternoon. My name is Simran Kaur, and I'm a student from Christ University, Bangalore. And today, the topic for my presentation is advancement
One
in.
minute. It is still loading your screen. It is still showing less loading. Yes, now we can see. Just present like this itself. Just move on with this.
Okay, sure, ma'am. So, good afternoon, everyone. My name is Simran Kaur. I'm a student from Christ University. And the topic for my presentation is advancement in the detection and analysis of novel psychoactive substances in postmortem toxicology. So, for the introduction, I would like to say what is NPS. So, NPS stands for Novel Psychoactive Substances, and these refers to the synthetic substances or chemical analogs that are designed to produce psychoactive effects that are similar to the controlled drugs. So, according to the reports, hallucinogens are the most frequently used NPS, that is with 73.8%, and that is followed by the stimulants with 13.9%, and then the synthetic cannabinoids 11.7%. So, the increasing prevalence of the NPS has presented significant challenges in the postmortem toxicology investigations, and hence an accurate detection and analysis of NPS in forensic context are essential for understanding the cause and the manner of death, as well as for the legal and regulatory purposes. So, this research paper will explore the recent advancements in the detection and analysis of NPS in postmortem toxicology, aiming to enhance the capabilities of forensic laboratories in addressing this emerging issue. So, for the beginning part, there will be an overview of the background and significance of NPS in postmortem toxicology, and with highlighting the risk they pose to public health and safety. The importance of accurate NPS detection is emphasized considering their diverse chemical structure and potential legal implications. So moving on with the literature review, as I already told that NPS refer to synthetic substances or chemical analogs that are designed to produce psychoactive effects similar to the controlled drugs. So about the classification of NPS, it is basically classified into 4 types. First one will be synthetic cannabinoids. These are chemical compounds that are designed to mimic the effect of cannabis. And they mainly attack, or they mainly act on the same cannabinoid receptor, that is THC, that is the active component of the cannabis. Talking about the synthetic cathinoids, these are also known as bath salts, and these substances are synthetic derivatives of cathinone, that is a stimulant found in the khat plant. And these typically produce the stimulant effect that are similar to cocaine. Third one will be phenethylamines. These category will include substances that are like 2CB, which are synthetic analogs of phenylphenamine, that is a class of drugs. And these have hallucinogen and stimulant, both the effects. Now, the last one will be piperazines. These are the group of chemicals that can have stimulant and hallucinogenic effects, and the substance like BZB will fall in this category. Now, talking about the analytical methods and the techniques that are already being used for the detection of these substances, the first one will be gas chromatography with nitrogen phosphorus detection. And this is commonly used for the detection of NPS, and this technique relies on the detection of nitrogen or phosphorus containing functional groups that are present in many of the NPS compounds. The next one will be TLC, that is thin layer chromatography, and this is mainly used for the preliminary screening of NPS. and it will involve the separation of compounds on a stationary phase that will be followed by the visualizing of the separated spots on the UV light or using some chemical reagents. The third one will be color test. This will involve the addition of specific reagents to the postmortem sample, which produce some basic color changes when they are reacting with the NPS compounds. Then the last one will be immunoassays. Immunoassays such as radioimmunoassays, that is RIA, and enzyme immunoassays, that is EIA. These are used for the NPS screening, and these methods will rely on the specific binding of antibodies to target some compounds. And these methods usually offer very rapid results, but they may lack the sensitivity because there are many advanced techniques that can be used. So the limitations of these techniques and the need for advancement. The first one will be rapid emergence of the new NPS. So the landscape of NPS is constantly evolving. So we need continuous method development and updating of the reference libraries that are already being used to ensure the detection and identification of the new NPS. The second one will be limited reference material. As I already said, there are very limited reference materials or standards that are already being stored, and the new NPS have been emerging. So we need some new standards that can follow up. Then it will be detection of low-level concentrations. So some samples, some NPS compounds, they would be present in very less concentration in the postmortem samples. For example, if the body or the dead body is found very late, so there's very less or contaminated samples that will be found. So achieving sufficient sensitivity to detect these compounds is very crucial. Enhanced detection capabilities such as low limit of detection and quantification are needed to identify the NPS accurately. So the methodology will be, the aim of this work is the development of a method able to detect, confirm, and quantify the set of NPS. Particularly, I have taken synthetic cations and phenylamines in the blood sample. So there will be a fast derivatization process in this because of the procedural complexity, and we need to do a derivatization process in the blood samples because it's a biological fluid. So the working solution is prepared with methanol at a concentration of 5 mg per ml, and these solutions are stored between 2 degrees and 8 degrees Celsius,
So
and they're
can
protected.
you move on to the result and discussion?
Okay, ma'am.
So after derivatization, how is it analyzed?
Okay, so the process is like the blood sample were diluted with 4 ml of phosphate buffer.
No, all these are okay. All these are fine. After derivatization, how is it analyzed?
So, ma'am, a GS analysis were performed on a mass selective detector with a capillary column. The injection was performed in a splitless mode at a constant flow of 1 ml per minute with a purified helium as a
Can
carrier
you go to
gas.
that results? Can you show those results alone?
Okay, ma'am, that is in the conclusion part.
Yes.
Yeah.
You can move on to that.
Okay.
No, any graph? You don't have any graph?
No, ma'am, I got the access to the machine right now, so I'm just continuing with the graph work.
Okay.
Yeah.
Okay then, thank you. So how many metabolites you separated?
Sorry, ma'am?
How many metabolites you separated?
So, ma'am, I took 8 samples.
Okay.
Yeah. So that is mainly the determination of 8 synthetic cations.
Okay.
So that is cation, flavidor, bofidor, alpha PVP, methylone, ethylone, pentilone, and MDVP. And including this, there will be 3 phenethylamines, that will be 4-MTA, 2-CP, and
The chromatogram as well as the spectrum is very important as far as the research is concerned.
Yes, ma'am.
Right? Okay,
yes,
thank you. We can move on to the next one.
Yes, ma'am. Thank you. We'll move on with the next presenter, David Devjit from Christ University for his oral presentation.
Good afternoon, ma'am. Am I audible?
Yes, you are.
Okay. I'll be sharing the screen. Is my screen visible?
Not yet. Yes, it is visible.
Okay. So good afternoon, everyone. My topic is a study on the diversity of ants, a new social insect, and their role in forensic entomology. It's a review paper which I had published. So I'll be talking on that, and it's related to my field as well. So I'm a PhD scholar from Christ University, and under the field of entomology. So this is the contents which I'm going to talk about, the introduction to the ants and their role in forensic entomology. Now, what is the forensic entomology? History, post-mortem interval, degree-day model, estimating PM, post-mortem interval by insect succession, stages of decomposition, geographical distribution, and seasonal diversity of ants, and how they help in forensic entomology, the research gap, conclusion, references, and acknowledgement. So introduction, coming to the point as why we use ants, like one of the insects, how ants play a role in the forensic entomology. Now, ants are the social insects, as what I said, and they, since a body, when it decays, it has 5 stages of decomposition. So throughout those 5 stages of decomposition, we'll be observing ants, and so it plays an important role. Now, the ants which are found nearby to the cadaver are Solenopsis invecta, which I'll be showing in the next slide. Solenopsis invecta, which can also be called as a red imported fire ant, the carpenter ant Camponotus rufipes, Pydol longipes, Hyponera, and the weaver ant we call as Ecophila smaragdina. Now, what is forensic entomology? Now, forensic entomology, when we come to the point, is that forensics. So it is the entomology is the use of insects and other arthropods in legal investigation, and to observe the measurable stages of insect growth and development. We can call it as referred to as forensic entomology. So how the insects can be used to solve a crime, we can refer to as forensic entomology. Now, the history of forensic entomology dates back to 13th century. So it was found when it was observed, and it was found when a farmer in China was found murdered by a sharp object, and the crime had all of the suspects of the crime had their sickles, and those were attracted to the blowflies. So that's what made it more curious and to more observe that the forensic entomology could be established. But until the 18th century, it was not found. But then in the period of 18th century, forensic entomology came up because of the fast-moving technologies and all. So now, as what I said in the contents, the post-mortem interval. Now, what is a post-mortem interval? So post-mortem interval is the studying of arthropods present in a corpse, which helps the investigators to determine the amount of time elapsed since the victim died. So suppose a murder has taken place, so the right amount, at what time the murder had taken place, we can find it out by following the forensic entomology procedures. So that we can call it as post-mortem interval. Now, post-mortem interval can be found out in 2 methods. The first one is the degree-day models, the first generation of insects, like the first colonizer, the first arrival of insects, the cadaver, we can call it as the insect colonizer, first generation of insects to colonize the corpse. The second one is the concept of insect succession when more than one generation has occurred on the corpse. So there's a wave of insect succession which occurs on the cadaver. One succession comes, and that succession leaves, and the other succession of insects, they arrive. That we're going to talk about in the next few slides. But first, we're going to talk about the degree-day models for the first generation of insects to colonize the corpse, how it is related to the post-mortem interval. Now, degree-day models, it can be applied when the first generation of insects, like the corpse, first generation of insects, which arrives at the crime scene. So in an experiment which was done by the University of Malaysia on monkey cadaver, it was found that within 15 minutes after death, a colony of Pydol longipes and Anglopolepis longipes, Phytoleucton diversus, Ecophilus maligna was observed near the cadaver. Now, the post-mortem interval, this process, it has a 5-step process which we need to follow to get out the post-mortem interval. The first step is to collect the arthropod colonized from the corpse. So all the stages of these insects should be collected because the age of different individuals can be difficult and they are different. So we need to collect all the species, all the insect species which are present, no matter which, like whether they are in the larval stage or whether they're in the adult stage. We have to collect all the stages of the insects. The second stage is species identification of the initial colonization. So then after collecting, we need to identify the species. What are the species that are present, like the ants or maybe the blowflies? What are the species that are present? The third one is estimate the development stage at which the insects were collected. So specific degree models are present in specific developmental stages of insects. So after finding out that we need to study the developmental stages of each insect. The fourth one is the collect short-term climatic data. Now there are few insects which are found during the particular climate, and those insects can be referred to as like it's okay. Maybe the crime scene had taken place during this season, and the body was found during the different in a different season. So by this we can find out that okay, the climatic data is also involved in estimating the post-mortem interval. The fifth one is the estimate the post-mortem interval. And the fifth and the last one is the post-mortem interval. By collecting all these data, number of degrees required, collecting all these data of the insect cycles, the insect stages, development, come in accordance with the climatic data, we can find out the post-mortem interval. Now, estimating post-mortem interval by insect succession, the second point. Now, since in a cadaver, as what I said, insect succession takes place. Like suppose one wave of insect comes, the second again they leave, the second stage of insects come. So how they can be measured? Now, in the next slide, I'm talking about the stages of decomposition. Here, I'm going to talk about the succession of insects. Now, here we can see that the stage of decomposition is divided into 5 stages. The first stage of decomposition is the fresh. It's known as fresh. So the first stage of decomposition where it occurs is that the body's immune system stops functioning and they release volatile organic compounds such as the cyclic hydrocarbons, oxygenated compounds, sulfur and nitrogenous compounds. So hence these organisms, they get attracted to these such compounds and they get attracted to the cadaver. So among these, ants, blowflies and other insects are found. The second stage is the putrefaction. So the second stage happens when this stage marks the arrival of the second wave of arthropods. So here the corpse begins to bloat due to the gas produced by the anaerobic bacteria, and we find that the first colonizers are accompanied by the second colonizer, that is the cheese flies and fanid flies as well. The third stage is black putrefaction. So this stage is characterized by the discoloration of the body and the bacterial decomposition. The flies, the first colonizers, now they leave the body, and the offsprings, except ants and beetles, they dominate the cadaver. The fourth stage is the butyric fermentation. This stage is characterized by, again, the discoloration of the body and escape of gases from the body as a result of bacterial decomposition. And the flies leave as it becomes unsuitable for them to develop their offsprings, and only ants and the beetle species, they dominate the cadaver. The fifth stage is dry decay. So here in the last stage is the dry decay, where only hairs and bones are only present and only the piece of dry skin is left. So most of the insects by this time, they have left the body, and except ants, beetles in the family, domestic, are only present, and they are the scavengers. So now the geographical distribution and seasonal diversity. Now, there is a lot of information as what I was saying, the geographical distribution. Now, there are some species of ants which are present in one geographical region, which are endemic to one particular geographical region, and some are not. So suppose a crime scene is taking place in a particular place, and the body has been dragged from that place to another. We can find evidences that by the insects, we can find that whether that species of ants is present in that body, and we can find that, okay, fine, maybe the crime scene had taken place in this particular region and the body has been dragged from that particular region to the other by the examination of the ant species which have been found, because they are endemic to that place. Similar to climatic seasons, climatic comparisons can also be we can talk about as it like some seasonal diversity. We talk like ants are found in some ants, like they're found during one particular season, and they're not found in the other particular season. So if a crime scene had taken in one particular season and the ants have been found in that particular season, we can conclude that, okay, fine, the body, maybe the crime scene had taken place in this particular season and not into the current particular season where the body had been found. Now, the research gap. Now, the research gap is that the ant species and the wave of insect successions, they vary across regions. And a model of insect succession should be developed for each region, like similarly for the degree-day models for insect development. And it can be used in forensic entomology, particularly for the initial colonies, the first wave of successions. So coming back to the conclusion, as like forensic entomology, as the days goes by, it is playing an integral part, and it has proved its feasibility in an investigation. Now, the degree-day models, as what I said, it should be with relation to the temperature, the climatic data also, and the age of the first generation of insects present on a corpse can be determined using degree-day models. Now, insects can provide information for forensic investigation beside an estimate of post-mortem units. So that's the treatment of a victim after death or the presence of drugs in their system. So insects can be collected and they can be analyzed to find out whether the drugs, whether the victim, whether the crime scene, whether the murder had taken place, whether drugs were present or not. And so this is what I want to talk about, and these are my references related to my work.
Okay, Mr. David, actually, it seems to be quite new field for us.
Yes, ma'am.
So is it actually used anywhere?
Yes, ma'am. Is it actually? Can you come?
Is it right now used anywhere? Practically, are they using it?
Yes, ma'am. It has been used, but in India, it will take some time.
That's what, because see, if you could have incorporated some of the case studies, then it would have been still interesting. It seems to be very interesting topic.
Yes, ma'am.
And another humble suggestion is your topic is fine. The volume of data you have collected is also very good. Your presentation is also good, but your slides are not that good. That is not adding to your presentation. Your background, the font size, everything has to be.
Yes, ma'am. Actually, too much of information.
Yes.
I had to add.
So you can concise and you can give it in a pictorial form, right? That attracts audience.
Yes, ma'am. Yes, ma'am.
Right, and please include some case studies. Let it be not necessary. It should be in India. It can be anywhere, so that the audience participants can understand in a better way. That's
yes, ma'am. Yes, ma'am. Sure, ma'am. Thank you, ma'am.
Thank you, Mr. David. We'll move on with the next presenter, Bhumika from Christ University.
Am I audible, ma'am?
Yes, Bhumika and Sujana, right?
Yes, ma'am.
Yes, you can start with your presentation. You're audible.
Is it visible, ma'am?
Yes, it is visible.
Good afternoon to everyone present here. My name is Sajana. I'm a student of Christ University. So our topic is quantification of drugs from hair, a review study. We all know that hair is basically a protein filament which goes from the follicles, and its function is like protection, regulating body temperature. But apart from that, hair offers also several advantages when compared to other biological samples in forensic investigation. So hair analysis is an emerging method to confirm the drug use in forensic investigation. And the most important feature of it is it won't actively metabolize the drugs once they're incorporated into the hair. And this incorporation can be done through passive diffusion from both blood and sweat. And once they're incorporated, they will be deposited in the keratin matrix and will be distributed along the length as the hair grows. And it is considered to be a better sample than the biological samples because of reasons like longer detection time and non-invasive and ease of collection. Analytical methods like GCMS and ICPMS can be used to identify and quantify the drugs present in the hair because of the reasons like these instruments have specificity, sensitivity, accuracy, and better detection limit. And these hair analysis can also be used to identify the doping agents which are used by athletes in Commonwealth Games. The athletes use these anabolic steroids to improve their body mass and strength. It will be helpful in testing and monitoring the drug use. The objective of our study is to verify the precision and efficiency of the analytical method, which is used to quantify and detect a variety of drugs in the hair. So the very important step is to collect the samples. So hair has to be collected from the vertex posterior, that is back of the head, where the hair will be in the same growth rate, growth phase, and it is least affected by age and sex of the person. So preferably it is collected very close to the scalp, and 60 hair samples has to be collected per sample. And why hair is preferred? Because it can retain the drug up to 90 days, which is suitable for the analysis. And after cutting the hair, which is very close to the scalp, it has to be wrapped in aluminum foil and stored in dark places under the room temperature. So we use scissors which are made up of polytetrafluoroethylene because these scissors will avoid the elemental contamination between the samples. And the next step is sample preparation. So it differs based on the instruments we prefer. So for GCMS and ICPMS, it is more or less very similar.
Madam, you can skip this and you can move on to the result and the actual work what you have performed.
Yes, ma'am. So GCMS, the method is a chromatography technique wherein we insert
this kind of introduction we don't want.
Okay, ma'am.
Share what you have done actually.
Ma'am, this is actually a review paper, so we have combined the analysis.
Okay. Any reported article on this?
Yes, ma'am. My co-author Bhumika will continue the presentation.
Okay.
So these are the results and discussions that we obtained from few papers. So the main objective of this study is to quantify the drugs from the hair. So commonly encountered drugs like morphine, cocaine, fentanyl drugs, opiates, amphetamines are detected using GCMS. So these are the general mass spectra of cocaine and morphine. So in mass spectrum, on the x-axis we plot mass to charge ratio, and on the y-axis we plot the relative intensity. So the peaks tells the fragments of the compounds present in the sample, and the highest peak tells the relative abundance that is there in the sample. So here is the mass spectra of lead and mercury that can be detected using ICPMS. This also works the same like GCMS. So the mass spectra here and shows the fragmentations of the samples or the compounds that is present in the lead. So this was our work that we've done, ma'am. So also we've mentioned the sensitivity of GCMS and ICPMS over other instrument. These instruments are preferred for analysis as it provides reliable data for administrative and judicial purpose. ICPMS is not only known for its sensitivity, but it is also has the capability to measure multi-elemental analysis. Hair is preferred because it is more sensitive than other biological matrices since parent compound can be detected only in hair. So this is the conclusion. To gain an understanding of the necessity of hair analysis in forensic investigation, hair processes unique characters that makes it preferable over other biological matrices. So additionally, the ease and non-invasive nature of collecting hair sample further contributes to its advantages as a preferred method for analysis. And quantifying hair matrix involves employing analytical methods such as GCMS and ICPMS. So these methods exhibit the capability to detect even minute quantities of the compounds, enhancing the sensitivity and accuracy of the detection.
Okay, what do you think are the limitations of these methods? You have told all advantages. I am asking the limitations of these methods.
If any contaminations are there, that might be a problem for the instruments to detect.
Okay, can you analyze all the drugs?
Not all the drugs, ma'am. Whichever drugs that are suitable for the instruments only.
That is again a limitation. Okay.
Yeah.
That is again a
It
limitation.
is not capable of analyzing the non-volatile drugs and polar kind of drugs. In
Therefore,
GCM.
this ICP-MS is not available that easily. It becomes very costly also, expensive, right?
Yeah, it's expensive, and only the elemental analysis can be done with ICPMS.
Yes, they are the limitations. Okay, good.
Yeah.
Thank you.
Thank you.
Thank you, ma'am.
Thank you, Sujana. We'll move on to our last scientific presentation, Vedashri from Christ University. Vedashri Pavana Sinchan Ullas Nayak.
Yes, ma'am. Am I audible?
Yes, you are.
Yes, ma'am. I'll share the screen.
Is the screen visible?
Yes, it is visible.
Good afternoon, everyone. Myself, Pavana MS, and my co-presenter, Sinchan Ullas Nayak and Vedashri D from Christ deemed to be University, Bangalore, will be presenting on a topic, toxicological profile of aspirin by UV spectrophotometer and FTIR, which is a review study. So basically, we need to know what is the toxicological profile. So it is a detailed description of a potential harmful effects of a poisonous substance on a living organism. So it basically involves the studying and understanding of how this chemical substance or a drug mainly affect the human health, and also profile which includes the toxicity levels of a particular substance, that is a chemical substance or a drug, and its various routes of exposures which mainly gives the long term or the short term health risks which are associated to the use or the exposure. And here our study mainly focuses on acetyl salicylic acid, which is also known as aspirin, and it is basically classified as a NSAID, that is non-steroidal and anti-inflammatory drug, and it is one of the oldest known analgesics, and it is originally extracted from the bark of the willow trees. And aspirin, it is majorly used in homicidal cases due to acute ingestion, and the lethal dose for this aspirin is 150 mg per ml, and when this toxicity increases in our body, it may lead to the death of a person due to pulmonary edema and also other organ failure. And therefore, in such cases, we need to detect and quantify Ma'am, is there any disturbance or? Okay, so therefore, in such cases, it is necessary for us to detect and quantify aspirin using various analytical methods. And here, our review study mainly focused on FTIR and UV spectrophotometer, where FTIR is mainly used to measure the aspirin and its major components, and UV spectrophotometer has the capacity to detect the molecule of interest. So my co-author will be dealing about the quantification methods.
Good afternoon to one and all present here. Myself, Sinchan Nayak, and I will be continuing with the sample collection as well as the quantification of aspirin using UV spectrophotometer. Now, we all know that before any quantification technique, sample collection is a major step. Thereby, aspirin can be obtained in the gastric content of any human being in the form of tablets or in the liquid form. Suppose we obtain it in a tablet form, we dry it, we collect it, dry it, and store it in separate containers. And during the experiment, we generally crush it using the mortar and pestle. The required amount is crushed and being used. First, the preparation. If it is a liquid sample, then we mix the sample throughout to ensure that there is uniform distribution of the active component, and out of it 30 ml of the aliquot is being stored in the plastic container. Now, how do we prepare the sample? First, the required homogeneous material is being dissolved in a suitable solvent, preferably methanol and ethanol, since they have good solubility rate. So this is the stock solution. This stock solution has a higher concentration with respect to the quantitative analysis. Thereby, we cannot directly use the stock solution. Therefore, we dilute it using the same solvent, example methanol or ethanol, to make the working solution. And the working solution is being used for the spectroscopic analysis. Moving to the spectrophotometric analysis, we prepare different standards of aspirin using different concentrations with the same solvent that is being used. And these standards that are being prepared, we irradiate it to the ultraviolet light, the UV light, with 200 to 400 nanometer range. So once they are irradiated, we get the spectra. In the spectra, we generally get the absorbance of the sample that the sample is being absorbed. So this absorbance, we generally record 3 absorbance, and the average of the same is being considered. To know the concentration of the unknown sample of aspirin, we use a calibration curve which helps in determining the concentration of the unknown sample. My co-author will be continuing.
Good afternoon, everyone. I'll be dealing with Fourier transform infrared spectroscopy. It is FTIR. We all know that its infrared spectroscopy is one of the useful ways for identification of drugs. It is basically an analytical technique which is used to determine the infrared absorption, emission, and reflection of a material. It is commonly used in most of the pharmaceutical industry, and commonly used technique which is used for quality assurance and quality control. This term Fourier transform infrared Fourier transform comes from mathematical process which is required to convert the raw data into actual spectrum. Coming to the instrumentation, it basically Fourier transform infrared spectroscopy. It consists of a light interferometer, sample holder, and detector. Here, the interferometer plays a major role, where it splits a beam into 2 parts, where one path will be passing through the sample and the other path will be passing through the reference sample. So basically, when
you can go on to the results.
Yes.
We don't want all this introduction.
Okay.
Yes.
Yes.
Yeah. So coming to the result part. As we can see, this is the resultant spectrum of UV spectrophotometer, where x-axis represents the wavelength and y-axis represents the absorbance. So as we can see, the characteristic peaks are being observed in the case of aspirin, and here the maximum absorbance range that is being maintained is 274 nanometer. And coming to the resultant spectrum of FTIR, here we can see the x-axis which represents a wave number and y-axis represents the transmittance, and the characteristic feature peaks are being absorbed in the case of the aspirin, and the maximum wavelength range of 4000 nanometer is being maintained over here. And this is the general representation of both UV spectrophotometer and FTIR that is being used. And to conclude our session, the aspirin, which is also known as acetyl salicylic acid, is one of the, it is considered as an over-the-counter drug, which is commonly available to all the people. So hence it can be easily used in any homicidal cases, and therefore it is very important for us to efficiently extract the sample and then to analyze using various analytical methods like FTIR and UV spectrophotometer. And these are all the references which we used to do our study. That's all, ma'am. Thank you.
Okay, why are you calling it as toxicological profile?
It is because it will mainly include the exposure routes and long-term and short-term effects, and how this affect, I mean, the particular chemical substance which mainly acts on the living organism, and its effects are being studied in the toxicological profile, ma'am.
Okay. Okay, madam. Thank you.
Thank you.
Thank you, ma'am.
You can stop sharing your screen. And that brings us to the end of the scientific sessions of day 2 of Pharma conference. I'd like to thank the Association of Pharmaceutical Research for organizing such a great and wonderful global pharma event. At this midst, I would like to thank Mr. Rudrabhanu Satpathy, CEO and founder of APR Association, for being the backbone of this conference and organizing such a worldly event. I'd like to thank our organizing committee members, organizing secretaries for rendering their active participation for the event. I'd also like to thank our all keynote speakers of day 1 and day 2 who made our day the knowledgeable one. And I'd like to extend my thanks to Ms. Ramya Devi, Mr. Abdul, and Mrs. Srilekha, who are the coordinators from the APR, for coordinating such a wonderful sessions and moderating the sessions in timely manner. I'd like to thank our technical team, who is the reason for this uninterrupted Zoom streaming and live streaming. And I'd like to thank our session chair of day 1 and day 2 for evaluating the sessions patiently by listening to each and every scientific presentation and asking the questions valuable. I'd like to thank our participants, listeners, and the presenters who have joined the day 1 and day 2 of the scientific session and made the day fruitful. Every ending is a beginning of a new beginning, and we'll be joining the next presenter. Thank you. And that brings us to the end of the day. Certificate downloading link will be sent to you via mail. Thank you everyone. Thank you for making the day grateful.